So there are three, and first talk is going to be Dr. Amit Yadav, and I would like to introduce Dr. Amit Yadav to this gathering. Dr. Amit Yadav is now working as a scientist in National Center for Microbial Resource, National Center for Cell Science, that is in Pune, and he did his PhD work that is on detection, diversity, and differential expression studies in sugarcane grassy shoot phytoplasma that he did at University of Pune, and he has got a very wonderful publications almost mostly on this phytoplasma. That is more than 54 publications he has got with more than 550 citations, and he is an editorial board member for phytopathogenic molecules. I think this is one of the recent journal a few years back. It was launched by Dr. G.P. Rao. I think it's a Springer publication. It's a wonderful thing, and it's very rarely we see the dedicated workers on phytoplasma, and I am very happy to welcome Amit Yadav, who is joining with our IRI team, Dr. G.P. Rao, and very few people are working on this very important obligate plant pathogen, which is almost ignored, especially in management and other aspects. And we are, I think all will be very keen to listen Dr. Amit Yadav. So I welcome Dr. Amit Yadav to go ahead with your presentation, and you can take not more than 20 minutes. Not more than 20 minutes. Dr. Amit, I think you can go ahead.
Yeah, thank you very much, sir, for the nice introduction. I was expecting to reduction in the lecture time, which is not very fair, by the way.
Speak, actually it is 30 minutes, sir. That too, we have less speakers.
Today's topic,
we have only four, instead of 4 we have 3
minutes we have. If you take 20 minutes, we will have 10 minutes discussion. If you take 20 minutes, we will have 5 minutes discussion. So please go ahead.
Right. Yeah, can everybody see the screen?
Yes, yeah.
Yeah. So today's topic is actually restricted to only diagnosis of phytoplasmas, strains associated with crops, wild plants, and insect vectors. The reason why I chose this particular topic is and restricting only to the diagnosis is because there are many queries come to me about the phytoplasma in those symptomatic plants or insect vectors. So I thought of restricting this talk only for diagnosis purpose. So diagnosis, before I begin, I think I must define again, though most of us are actually belong to pathology stream and no need to actually really redefine diagnosis here, but just to make sure that everybody is on the same page, there's a difference between detection, identification, and altogether comes down to diagnosis. So diagnosis doesn't mean only getting an amplification of phytoplasma or for that matter, any pathogen. Diagnosis doesn't mean that only identification of a particular pathogen. Diagnosis means that the symptoms that you see on the plant or any particular specimen, you come to a conclusion that which pathogen, including its taxonomy, that what is associated with that particular disease. So I'll be taking you through the little minor part of the protocols that I follow, or rather we follow in our lab, and some hints, some tips, some precautions that one need to, or considerations that one need to make while detecting phytoplasma in plants or insect vectors. Again, I think I will not spend much time in telling you that what are phytoplasma pathologies, because the audience is all belong to pathology stream. I will not spend much time on this. Also, there are some symptoms. Let me just take you through all possible symptoms that are seen
Your voice is breaking, Dr. Amit.
Different plants cause, though we always In case of phytoplasma, because cross postulate is not possible, you cannot really inoculate phytoplasma.
Sir, Amit sir, your voice is breaking. Your voice is breaking.
And have a caution. Okay. Well, everything is intact. I don't know why.
So is it, no, your internet connectivity is okay? But Amit, can you see your internet connectivity?
Yeah, that is perfect.
Perfect, no? Then what you can do is you can put off your video. You can put off your video and then
1 second. Okay, can you hear me by just removing the earphones?
Now good. Yeah, it's very good.
Is that the reason?
Yeah, it's good now.
Now good. I think the earphones were the issue then, probably.
Yeah. Please go ahead.
Yeah, please do if the audio is not good. So telling you about in case of phytoplasma diseases. So it is always the disease is associated with phytoplasma, and particular phytoplasma is related to that disease. So related to and associated is the word which is always used, and not caused by phytoplasma, or other terms are used. So these are the different symptoms which we see in case of phytoplasmas. What do they cause? And there are terminologies which is used called blooming, or little leaf, leaf yellowing, or whitening, or there is another term which is called as fasciation. If you see on the left hand side is a Cestrum nocturnum plant, the middle one is Acalypha, and the last one is a Valencia, or the old name is Acacia. And these are some of the pictures which are taken from the internet, which is one of the most common symptoms which is seen in case of phytoplasma, is phylloidy. So these are some of the pictures that we are working on. These are some of the example pictures that we are working on. This is the first one is the SSD, sandalwood spike disease. And if you see, recently we visited Marayur. We are working there. We have got extramural project on this to work on to find out the vectors of sandalwood spike disease. And this is how the tree look, completely a dead tree, very scary image. And the typical symptom that we see normally is the spike. And I think also most of the people are aware of these symptoms. In another project that we are working on is the soybean phytoplasma, and this particular disease is called no pod disease. Because it is related to phylloidy, there is no flower development. So obviously when there is no flower development, we don't have a pod formation there. If you can compare the pictures against the first picture of a healthy plant, you will see that the phylloidy causes no pod disease. This is another plant which is related to forestry, because the sandalwood phytoplasma we are working on is in with collaboration with IWST Bangalore, which is related to wood science, and they are related to forestry. So this is another important tree as far as the social forestry in Maharashtra is concerned, because there are a lot of acacia catechu plantation has been done all over Maharashtra, particularly in Maharashtra. And this is how see on the roadside is Ficus amplissima, and the middle one is the healthy plant, while the rest two are actually the symptomatic plants. If you are passing by, passing through, or passing beneath the tree of a ficus and it is symptomatic, this is how the plant look like. And this is the one are actually witch's broom plant, and these are the different plants showing symptoms. These photographs were clicked in around Pune plantation. One of the most common phytoplasma seen in Ziziphus. This is wild Ziziphus called Ziziphus oiniplia. And if you see on the extreme right is a healthy branch, while the rest two are actually the infected one showing leaf yellowing and the little leaf. Another common phytoplasma seen in sugarcane, where you have and the disease is called as grassy shoot disease, because the symptomatic plant looks completely looks like a grass. This is the example that I definitely want to show all of you. Is the Tamarindus yellow leaf phytoplasma. Now, anybody passing by this particular plant may not realize that this is a phytoplasma infection, because you will only see that one particular branch is turning, the leaves are turning yellow. This is another photograph that I have taken. This is the third one. But if you see particularly, unlike other plants, they were showing symptoms of either changes in the morphological changes seen in the different organs. Here, there is no particular change in the morphological structures, but only color differences. On the right hand side, you will have a healthy plant which is completely lush green, while the on the left hand side you will have a infected plants can seen only with the changes in the color. So we are working on many different plants, almost 50 odd different plants we are working on today in our laboratory, ranging from various crop fields, insect vectors, and as well as many wild plants. Also now has underlined for a very important plants for the as far as the forest is concerned. Because today's topic is about the diagnosis of phytoplasma, I'll just browse you through what are the detection methods which are used in general, but of course PCR remains the primary one. This is for the information purpose that the fluorescence microscopy is used for detecting phytoplasma, and you can use the DAPI as a stain. But again, it is very laborious and it is not very conclusive method to say that the plant is infected by phytoplasma. Another one is transmission electron microscopy that one can use, but this is also very laborious, very technique oriented. You must have a transmission electron microscope in your institute or in your city at least, so that you can actually go and screen multiple samples. Again, not very good method for very large number of samples, and it can be only studied for some structural parts of the phytoplasmic bodies seen in them. Not very much recommended. Fish fluorescent in situ hybridization, where you actually design a nucleotide probe tagged with some fluorescent protein, and you can actually hybridize your tissue with these ones. Again, not very recommended technique, because it's the reason one is the fish probes are expensive, and the overall procedure is also quite laborious and time consuming. But one of the most important advantage of this particular system is that if you want to locate phytoplasma bodies in particular tissue, like for example in insects or in a particular organ, which are, for example, we are using the fish now in trying to understanding whether the phytoplasma are transmitted by seeds or not. So we are actually have designed a fish probe to understand whether in which part of the seed the phytoplasma is located. And there are many other techniques which I will not go into very much detail. There are serology based techniques, ELISA method. There is a heteroduplex mobility assay. There is a TRFLP, SSCP, and also the LAMP, which is also used by some workers on the field. All these techniques are reliable, but again, not very much used or not very popular among the scientists or in the extension scientists also. So the most reliable techniques which is used all over the world, which is conclusive for many different purposes. It can reliably detect the phytoplasma. At the same time, it can also lead to identification of that particular phytoplasma by including the sequencing of the target tissue. So P1 and P7 are the most common primers which are used for detecting the phytoplasma, followed by the nested PCR primers R16F2N and R16R2. These are the most common used primer paste used for detecting the phytoplasma, and these are developed quite almost 20-25 years back. So what are the different steps which are involved in these ones in detecting the phytoplasma? So obviously it will start with DNA extraction followed by the first round of PCR by P1 and P7 primers, second round by F2 and R2 primers. Then the application, the amplicon that you have obtained, you have to go for a sequencing. Then you have a sequence analysis. Then you have a sequence similarity searches on different databases. Then you can also go for a subgroup assignment using for that particular sequence, and the phylogenetic analysis if it is required. Now, before I go to the sequencing part directly, I must rather take you through some of the queries that we receive in our lab, that why a particular sample that we receive do not really amplify the phytoplasma despite it is having good symptoms in it. So there are many different considerations that you might have to do because we worked on array of different plants, right from the very simple, you can say the Parthenium, simple in terms of its content and the amplification efficiency in these plants. So the most difficult plants that we found are the woody plants, where the phytoplasma titer is spread all over the plant, or maybe located at one particular place. Especially in woody plants, these phytoplasma located at the lateral meristem issue. So you have to be very careful while choosing the tissue also, as far as the woody plants are concerned or the big plants are concerned. Herbaceous plant, obviously the phytoplasma spread is somehow even, but if you choose the floral part or the phylloidy tissue, that will give you more reliable amplification results. What we found is, instead of using plant tissue directly, some people use plant sap. A very conventional method of liquid nitrogen crushing followed by CTAP method is the most reliable method for detecting phytoplasma. If, like for example, in tamarindus or in soybean, we have very high content of polysaccharide in case of tamarindus, or very high protein content in soybean, we gave, obviously, with in case of soybean, we treated the DNA with more of a proteinase K to kill those proteins, or in case of tamarindus, we wash the DNA several times to get rid of the extra polysaccharide. In many plants, however, we have some compounds which can be removed by activated charcoal. So that we have used, but you lose a lot of DNA in due course. So proceed with caution if you have good amount of DNA, or you can aliquot the DNA and then give charcoal treatment to remove some of the secondary metabolites which act as a PCR inhibitor. That can work. We also use increased use of PVP because PVP is already there in the CTAP standard protocol, but increased use of PVP also works many times to keep the DNA in an amplifiable condition. And use of commercial kits. It is not necessary that all commercial kits will work on all possible plants. No, we have seen that in woody plants, or in where you have a very hard tissue, or the one which is having very high content of secondary metabolites, the commercial kits do not work very well. And once the DNA is out in commercial kits, you really cannot treat the DNA also, because the DNA is fragmented one, and the shelf life of that particular DNA using commercial kits is also not very long. So we relied, the bottom line is, we relied more on a liquid nitrogen crushing followed by the CTAP method, and trying to keep the secondary metabolite at the bay as much as possible. Second consideration about the DNA template, the DNA that you have got. Now, there are a lot of chances that even if you have a DNA and you cannot really treat more beyond a point, you take as less as DNA as possible in your PCR assay. So the notion of taking more DNA so that you will have more possibility of amplification is wrong. So you must really standardize the DNA content as far as based on your plant, that how much DNA you have to take, but lesser the DNA that you take, you are less contaminating your PCR assay with secondary metabolite or any other PCR inhibitor if it is there. So the bottom line is that the quality of DNA template has to be good. It should be PCR free. And even if there are some inhibitors which will remain, we cannot remove beyond a point, you have to use BSA, betaine, or DMSO. So in our case, in most of the plants, we have good success in using BSA as a PCR additive, and it gives reliable amplification. Another part is that choice of TAC polymerase. If you use a synthetic TAC, which are quite cheaper one, and we normally purchase these TAC synthetic TACs. But what we have realized that if you use high fidelity TAC which will work with little difficult templates coming from different plants or insect vectors, particularly insect samples also, they work well with amplification of phytoplasma. So that was one point where we realized for sure that the synthetic TAC or the normal TAC is not very efficient in detecting phytoplasma or amplifying phytoplasma target. So I definitely recommend it to use. It is costly affair, but it's more reliable in using high fidelity TAC. Another important aspect of using high fidelity TAC is that the sequence quality you get later on. With normal synthetic TAC, there are a lot of chances, or rather there are chances to have biases of adding a nucleotide. So that goes off, and high fidelity TAC will have can assure you the good sequence quality at the end. The use of bacterial universal primers for sequencing is something that I want to talk a little more in detail in next few slides. And I have seen that many people use only phytoplasma specific primers in amplifying the phytoplasma sequence. That's really not necessary. You can ease that by using bacterial primers. Now this is how the primary PCR P1 and P7 look like on your left hand side, both the images. And many times it is not amplified, so you can only see some smear or something like that. What happens in nested PCR is that you get some good amplification, and that is where you will have your phytoplasma confirmation done on the gel pictures. Now, if you see these pictures, there are many samples, even though they were symptomatic, there is no amplification is happening on all the three gels that you see on the right hand side. So what we did is, because I told you already that these primers are designed in early 90s, almost now 30 years back, we redesigned the nested PCR primers, keeping the P1 and P7 same. And if you see on the left hand, on the right hand side, the kind of amplification efficiency that you see as compared to the gel pictures on the left hand side. So we have used these templates which are coming from the primary PCR and diluted them with 5x, 10x, or 15x, or 20x. But we have results only for few samples turning positive. When we use these newly designed primers, you can see the same set of samples. They have amplified nicely, and now we are using it very routinely, and we will be publishing these primer pairs soon for the phytoplasma workers, so that they will have efficient efficient amplification of phytoplasma. So once you have the amplification of phytoplasma, in many other presentations, I have shown that how the sequences from Indian fraternity, those who are working on phytoplasma, are actually not giving the good quality of sequences. There are hardly any sequences which are, first of all, full length sequences, which can be used to assign these sequences to some taxonomy of phytoplasmas. But then I thought of having this particular picture where you have a 16S RNA gene, and now P1 and P7 are located on the extreme of the 16S RNA gene in this particular picture. And now you can use either of these primers to sequence your the 16S RNA gene. So it is not only for phytoplasma. Even if you're working on any particular bacteria and you want to identify, you want to sequence the 16S RNA gene of that particular bacteria, you can use these primer pairs to amplify, to sequence the 16S RNA gene. So what happens when you use 4 primers? You can have a 2x coverage of a particular region. So you're actually necessarily sequencing a particular region twice by different primers. So this particular information is available on Silva database. If you go and see, you have a lot of information given on how to use, which primer to use, and which primer to use for sequencing. Normally, if you use internal primers, internal primer, I mean to say P1 and P7 or F2 and R2 are the terminal primers, right, which have amplified the target. If you use the internal primers, like you can see this particular image, your sequencing efficiency will be good and the sequence quality will be good. Another problem that I see in many sequences which are there on the databases, particularly from India, are errors in the sequences. Now, the first image on the top is a good quality sequence, though there is some minor contamination you can see at the bottom of it. But there are many erroneous sequences submitted as is. Now, these can be detected like this. So if you normally any Sanger sequencing method that you use, you will have the first few reads, almost 20-25 base pairs, are bad quality. You need to actually remove them and use these reads read by other primers. That's the advantage of using the bacterial primers. So if you submit the sequences with the errors, your sequences will not be get cited, and they will be kept aside. So many people use NCBI BLAST to identify which the closest organism, the closest phytoplasma, by using 16S RNA. There is no harm in using, of course, NCBI BLAST, but the problem is that it is not dedicated database. So what we use is there is a database called Easy BioCloud, which is dedicated only for microbiology, all bacterial sequences, bacterial genome sequences, as well as bacterial 16S RNA sequences. So when you search your sequence against the database which is here, which is highly curated database, you will have more reliable identification done for your phytoplasma there, or for that matter, any bacteria. The subgroup classification, which is now getting a little outdated, and people are started relying more on the genome sequences. This is the home page of the portal, which is used for phytoplasma group assignments, group or subgroup assignments. But only important thing that this will work only when you have taken care of if you have taken care of your sequence quality. If the sequence quality is not good, this portal is likely to give you erroneous results. Okay, so this is how it gives the results, and we have the phytoplasma we found it associated with Ziziphus and Ficus are actually new subgroups. We also did the actual RFLP to see and verify whether the sequence quality that we have got is actually whether it is really working or not. So we did that particularly in this one. Now, not relying only on 16S RNA based identification, one can also go for MLST. So MLST is multi-locus sequence typing. So the typing is the one which is where we use where the word is used only for the declared scheme for multi-locus sequencing. In case of phytoplasma, we should use the word multi-locus sequence analysis, where we have multiple targets of a gene which will amplify and sequence, and then we choose a phylogenetic analysis based on that. So one of the examples that I can give you is the Indian isolates which are related to PWP, peanut witches broom phytoplasma, whether they belong to Australisia or Candidatus phytoplasma or Orentifolia. So what we did the 16S RNA gene phylogeny, where it is not very much clear that to which phytoplasma it belongs to. Then we gone for MLSA, the multi-locus sequence analysis, where we amplified 5 genes, out of which I have shown you 3 of them here. The first one is Lewis, second one is SECA, and the third one is EFTU. Now these are the 3 genes which are most routinely used nowadays to identify or delineate the phytoplasma species Now, another example that I can give you is the whether the Indian isolates which are related to sugarcane phytoplasma, whether it is Oryzae or Cyanodontis. Now, again, 16RNA didn't resolve too much. At the same time, SECA and the EFTU phylogeny also didn't resolve or didn't shed light in understanding whether it is related to Oryzae or Cyanodontis. So what we did is we actually gone for genome sequencing of the sugarcane phytoplasma. And the most close relative of sugarcane phytoplasma is sugarcane cyanodontis. It's a candidatus phytoplasma cyanodontis. So we sequenced both these phytoplasmas and then drawn a phylogeny tree based on genomes. Now, what happens here is that you are not relying on one single marker or even 2 or 3 or 4. You are actually taking the information right from the entire genome. So what we did here is that the first set of phylogeny was which was based on. Now, if you see here, there are 2 bootstrap values which are shown in the tree, which is based on 2 different methods. One is called as BPGA and UBGC. I will not go into details. Maybe one can write me back on asking what these are. But the BPGA is based on 10,000 odd amino acid positions, and the UBGC is based on 86 plus 1,000 base pairs. Marker genes coming from these genomes. So you can see that the tree is based on humongous data, and nothing really can now change this particular tree, that how it looks like. Now, to solve the query whether the sugarcane phytoplasma is whether it is Cyanodontis or Oryzae, it is neither of it, and we recently published one paper and we named sugarcane phytoplasma as Candidatus phytoplasma secari. This is another method that now going reverse, that we used to go for a MLST by amplifying the genes and designing the primers for that particular gene. Now what we have done is we have fetched the sequences of these particular genes from the genome itself. Now redesigning the MLSS scheme based on genome sequences, and we have used 3,700 amino acid positions to draw this particular tree. And this is also quite stable tree. If you can see the bootstrap values, which are very close to 100, and nothing can be really problematic in this particular tree. So thank you very much, because this was only related to the diagnosis, right from the single locus to the genome based one. And we also had a time constraint to present everything in detail. So I thought, let me just take you through what we have done and how the diagnosis has been done in our lab. So right from the 16RNA to some genes to genome sequencing, and we have also published one paper on one new species, the first species of phytoplasma from India, and that is associated with the sugarcane phytoplasma. Thanks a lot.
Thank you. Thank you, Dr. Amit. It's a wonderful presentation. I am listening to a phytoplasma exclusive presentation, and in fact, you have covered a vast number of phytoplasma occurring in forest trees, tree crops, and also the cultivated crops. And it's very, very nice talk, especially the point of diagnosis and as well as characterization. We can take 1 or 2 questions, very quick questions. If anyone wants to ask question, immediately you can come and ask, or you can put it in a chat box and we can look at the end of the presentation. Any questions, please? You can raise your in the reaction box. There is a reaction box. You can raise your hand, like here.
Hi, Dr. Amit. This is Ravjit Khengura. I'm from Perth, Western Australia, and thank you for your very nice presentation.
Thank you.
To mention that fasciation is also caused by phytoplasmas.
Sorry?
Fasciation.
Yes.
So we see quite often in canola in Western Australia.
Right.
Some species have more fasciation than others.
Right.
So do you think it is caused by phytoplasma in Brassicariopus in canola?
I have heard about phytoplasma infections in canola, but until unless I really see or see the pictures of those symptoms, I will not be able to comment, because many phytoplasma symptoms are also very close to viral symptoms, or many virus diseases also are much I mean, they develop symptoms caused by phytoplasma. So one can not really conclude whether it is phytoplasma or not. I need to first see the pictures, or you need to really run the PCR for that one to check whether it is phytoplasma or not. But I think that initially, prima facie, if you can show me the picture, maybe I'll be able to comment on it.
Okay, that's great. Thank you. I'll send you some pictures.
Thank you. Thank you. Dr. Amit, can you close this PowerPoint?
Yeah, 1 second.
Sharing. Yeah, just, yeah. The sharing, you can just stop sharing like that. You can stop sharing.
I think I have stopped it. I don't know.
Is it?
Because it says resume share. So
no.
I have already stopped it. 1 second. I'll quit the meeting and I'll rejoin.
No. Otherwise, we can ask the secretary to stop the sharing also. Someone can do it. Say, sir, can you do it? Okay. Are there any questions? Because there is a time constraint. I have got only one question, Dr. Amit.
Sure, sir.
See, you know, whenever we go for detection, we see typical symptoms of phytoplasma. Then we go for amplification, and we are identifying which phytoplasma it belongs based on amplification, sequencing, and etc. But the point is, can we do this detection when the plant does not show any typical symptoms? Right, the plant looks healthy, like, but it has a phytoplasma in it before it is manifesting into a big symptoms and all. Such experience do you have?
Very much, sir. Very much. I mean, this is one of the aspect, apart from insect vectors, when we know that they transmit the disease, we are actually finding out another ways of transmission of this particular disease, and the one is the seed transmission. I'll come to your point in very short. So what happens is that because the seed contains very low inoculum, and seedlings or even the hardened seedlings may not show the phytoplasma symptoms. That is one channel that we have found. We have taken the entire tray. I will not disclose the name right now, and checked all the seedlings for checking the phytoplasma. And we found, though the percentage is not very high, we found there are phytoplasma positive plants, and though they were completely asymptomatic. Another study that we did long back in sugarcane, where we took random samples from the sugarcane field, those who are symptomatic and asymptomatic ones. And we found there are asymptomatic plants which are harboring the phytoplasma in a very low quantity. Now, what we are doing now is that we are checking these plants by real-time PCR, and really understanding that how much is the inoculum threshold which should be there, or which is normally required in that particular plant, by calculating it, how much is the titer which is available over there, which is present over there.
Okay, thank you very much, Dr. Amit. Now we'll go for the second speaker. Can you this screen has to, now it's okay, I think so. Yeah, now we are closed. Now I think the second speaker is Dr. Hema. I think Hema is here.
Yes,
okay.
Sir. Yes, sir.
Okay, so yeah. I would like to just introduce her, Dr. Hema. I think most of the people who are all here listening to the talk might be knowing about Dr. Hema. He's a well-known virologist. Now he's professor and head Sri Venkateswara University, a well-known virology center in Andhra Pradesh, in Tirupati. And you know, and she is one of the student of Dr. P. Srinivasulu, the late Professor P. Srinivasulu, and she did her MSc in Sri Venkateswara University, PhD also in Venkateswara University. Then she was having a PDF and worked at Texas A&M University for a PDF. Then he joined Indian Immunologicals Limited for 2 years. Then she was assistant professor in biotechnology, Sri Padmavati Mahila University. And also she had an DBT overseas associateship, and she visited USA for 2 years for undertaking that work. And she joined again to the virology SV University as an associate professor in 2009, and now at present she is a professor. And very, very great thing that she has got 5 patents in her name as a first inventor. And one is from India, and another 2 she filed a patent in USA. Australia and Europe, each one there. Then she has got 60 wonderful publications, highly reputed journals, and she handled six projects sponsored by UGC, CSIR, and DBT, worth of 2.5 crores. And she's also a Fulbright Nehru Academic Professional Excellence Fellowship she received, and she worked at Case Western Reserve University, USA. And again, the European Erasmus Mundus Brave Visiting Professor she received, and she is a fellow of Andhra Pradesh Academy of Sciences, Amravati. And I know Dr. Hema, since she has got one paper on cucumber mosaic virus from Indian Institute of Science. She worked way back in end of 90s, I think so. And I read her paper, and I used to read a lot of papers on her work. And I welcome Dr. Hema, and you can go ahead with your presentation. And I think I would give you 25 minutes. If you quickly finish, we can have good discussion. Thank you.
Thank you, sir. Thank you for the nice, kind introduction. It's indeed a privilege and honor to present in this prestigious symposium, IPS Central Zone. And I thank the organizers sincerely for the opportunity. So really, it is a wide topic which is really difficult to cover, but I would try to do that. And the characterization, diagnosis, and detection of plant viruses. So I'm going to take a case study for that. So in this presentation, I'm going to give you only overview. I just want to submit at this point that because I prepared this presentation mostly for the students' sake. So I would cover most of the major highlighting points only. I would not go into the details of each and every technique because it is impossible to cover. So I would like to take a case study, though it is very old study, but it holds very good with respect to the title and topic. So my own experience, I'm going to share it while doing the characterization of viruses and the detection of viruses. So Dr. Amit made my presentation a little easier by telling the difference between detection and diagnosis. So I would just start with the presentation with saying that as you know that the viruses are like second important pathogens after the fungus, and they cause huge economic losses with respect to many of the important agricultural and horticultural crops. And a lot of symptoms they produce on the plants, and apart from that they cause a lot of problems with respect to the yield of different products of the crops also. So some of the symptoms I've shown here, and some of the viruses shapes I've shown here, that these are all the causal agents to cause those important diseases, and there is a huge complex interaction will be going on to cause the disease. And so just to give you a brief idea about the virus structure, as you know that there are very simple entities, just a capsid protein and a nucleic acid in most of the viruses, and in some of the viruses they will have an envelope. And the classification, if at all, if you look at it with respect to the ICTP, then you will see a lot of number of viruses which has been categorized under different families and the genera. So the question is now that when you get into the characterization of plant viruses, then you would be wondering that how people are categorizing these viruses, the viruses which are not known, the viruses or which are new, how they would be categorizing those viruses into these groups, or any new group which is going to come into join into this kind of table is something which we are going to deal with now. So why most of the times the identification of plant viruses is not easy, because as you know that just now Dr. Amit also said that the symptoms are very, very confusing. Sometimes you will be phytoplasma symptoms also will be looks like virus symptoms, and they're very complex, both diseases, and many strains will be there for one virus, and one strain can have different symptom expression in different varieties, and several viruses can occur at one same time, and the interaction between virus and vector is complex, and it can be confused with nutritional deficiencies and herbicide damage and phytoplasma diseases sometimes. And So the characterization of plant viruses per se, if you look at it, so it is nothing but the description of a range of properties of the viruses. As you know that the virus consists of a simple protein and the nucleic acid. So when you describe the characteristics of protein and nucleic acid, and you will be able to identify the whole characteristics of those two entities, then that would come into the characterization. So then those properties, when you do that characterization, then it will give you an idea about the taxonomic identification, and which is very, very important. And why we need to do this characterization, there are umpteen number of advantages for it. And as such, the next topic, that is diagnosis and detection. So both go hand in hand actually. Without characterization of viruses, sometimes it is really difficult to diagnose the virus, because so you need to have some of the information about the virus to get into the ideal detection technologies. So to develop diagnostic tools, you need to have some idea about the characterization, and to understand the genome characterization to get into the taxonomic status. And the organization of polypeptides and virions are very important to develop gene vectors, and to exploit the virus genes and sequences for production of transgenic plants. So now this is a new emerging area where I've got the Fulbright experience, where plant viruses are used, both VNPs and VLPs are used for some of the biomedical and biotechnological applications. Very, very interesting and novel area, virus-based nanotechnology applications. So for all this, if you have some kind of idea about the characterization of virus, then everything is possible. So for that, some of the characterization of plant viruses, it needs to have the purified viruses. So you cannot do all the characterization by using the crude sap or the semi-purified preparations, because you may not get that much good results. So when do you need purified viruses, like to produce polyclonal antibodies, of course, and then for some of the physical properties and biochemical properties and molecular characteristics, you might need a homogeneous preparation of viruses, which are very, very essential. So purification, just to give you a glimpse of it, so generally you need to maintain the biologically pure virus. With respect to plant viruses, it will be local lesion assay, and you need to have a suitable buffer and clarification, concentration, and further purification by centrifugation, generally the density gradient centrifugation, and do a final pelleting. So with all these steps, you might end up with a purified homogeneous preparation of a plant virus. But you need to, though it looks so simple, you need to have a kind of really a standardization to get into a point where you get the light scattering zone. So this is what I want to show you, that the virus looks like a band wherever the density of the virus is equivalent to the density of the sucrose. It has got banded there, so you will see a light scattering zone there when you put the light on the top of the tube. So with respect to the characterization of viruses, as I said just now, that I'm going to give you only the list of characteristics. So biological properties, if at all, if you look at it, then you need to consider the host range, both natural and experimental host range of the virus, and pathogenicity, association with the disease, and tissue tropisms, pathology, histopathology, mode of transmission in nature, vector relationships, cytopathology, inclusion bodies, sometimes geographical distribution also, and cross protection. And most of the times the host range itself is not enough. People are not able to identify some of the new infections based on the biological properties alone, because sometimes it is confusing and it is not giving a authentic idea. And antigenic properties, which are mostly related to the serological relationships and mapping epitopes. And with respect to the bigger family like Potiviridae, they found that it is very, very confusing to look at the serological relationships because it's a big mess altogether. So this also is not working. This considered as a little conventional, but it still holds good for many of the important virus characterization. And third category is virion properties. Under virion properties, you may have to look at the morphological properties of viruses: size, shape, presence and absence of envelope peplomers, and capsomeric symmetry and structure. So you will have different techniques altogether to look at all this, and physical properties where you look at the molecular weight of the protein and ion density, sedimentation coefficient, pH stability, thermal stability, cation and solvent stability, detergent stability, and electrophoretic mobility. So this all will give an idea of the virus stability altogether, and then some of the important characteristics. And then properties of proteins also, you can look at it: number, size, and functional activities, and amino acid composition, and sequence comparisons. So I'm going to show you an example where we come to a conclusion by looking at all these properties, and it would be very interesting by looking at it. So then properties of the genome is ultimate and final, and where you can get a final conclusion of the taxonomic status of the virus, and where you will look at different properties altogether. Again, the type of nucleic acid, whether it is RNA or DNA, and strandedness, whether single stranded or double stranded, linear or circular, sense of the viral genome, and number of segments, size of the genome and genome segments, presence and absence of caps, and the modifications at the ends, and polytails at the end, and nucleotide sequence comparisons are the ultimate criteria where you can get into a conclusion that this virus belongs to this group or this genus, something like that. And if at all, if they are enveloped viruses, then you may have to look at the presence and absence of those important lipids, carbohydrates, polyamines, cations, and enzymes also, and nature of them also will give you some kind of idea. So this is just a general overview about the characterization of plant viruses, where you consider all these properties and try to look at, you may have to look at most of the properties, or if it is possible, feasible at your laboratory to look at some of these characteristics, then you can just get into a idea. So if not, you have to go for a final conclusion, that is nucleotide sequence comparisons. So then fourth category is genome organization replication. Once you have the complete genome sequence, then you may have to look at the organization of the genome, how many ORFs which are present, what are the genes which are coded for what kind of proteins, and strategy of replication is much more deeper study, and characteristics of transcription. So where are the promoters, how the polymerase is binding to the promoter, which are the nucleotides which are important for the binding of the polymerase to the promoter. So characteristics of translation and post-translation processing, how the viral genome is making use of the host machinery, how it is able to get the host machinery to be diverted to the replication of its own genome. Everything comes under this category. And sites of accumulation of viral proteins, site of assembly, site of maturation and release. So all these characteristics will lead to a complete story about that particular virus. So now I'm going to quickly go through a case study by taking our own lab experience. So where we try to characterize a virus which is causing the mosaic disease of sugarcane, because I've said here as a virus causing the mosaic disease of sugarcane, because at that time we really don't know which virus is causing this virus. So we don't know the taxonomic identification of this particular virus which is causing the mosaic disease. So we thought that initially when we started the study, we thought that it could be a potty virus which belongs to the sugarcane mosaic virus group, which used to be very prominent and which used to be the causal agent in all the other countries around the world. So when you just get into the details, so I don't want to go through the importance of sugarcane. You all know how important it is. So India is very important with respect to the commercial sugarcane production. So when you look at the viruses which are infecting the sugarcane, so it used to be sugarcane mosaic virus all the time, and there is one more virus, sorghum mosaic virus. Both belongs to the genus potty virus. So people were thinking that the potty virus, so either this virus or this virus is the causal agent for the mosaic disease which is causing the which is infecting the sugarcane in India also. But we came to know that after our study that it is not sugarcane mosaic virus, it is sugarcane streak mosaic virus, which belongs to altogether a new genus, Poaceae virus in the Pottiviridae family. And this has been proposed based on the whole sequence of whole genome sequence of this particular virus. So I will show you how we get into that. So the general information, so when we want to look at it, so we tried to look at the literature. So we found that these are all the different details which are present with respect to sugarcane mosaic virus, but based on only the conventional biological properties and the little physical properties, but no molecular criteria has been used for this. So A to N strains were reported only based on the biological characterization. So to just give you an idea about the Potiviridae family, it is the largest family where you have the 200 plus definitive members, and it causes greater crop losses when compared to all the other families in the plant virology. And then so you have altogether 10 genera in this as per the ICTV 2019, and there are 3 species which are unidentified. So when you look at all the 10 genera, this is the Poaceae virus is the one which has got the Triticum mosaic virus as type member, and sugarcane streak mosaic virus belongs to that. How, I will tell you now. So this is the general characteristics of Potiviridae, where the most important part you need to remember is that cytoplasmic cylindrical and pinwheel inclusions is the characteristic feature of the Potiviridae family, and flagellous filamentous rods, of course, and single polypeptide and single stranded RNA, generally monopartite, except for the biomoviruses. So genome organization, it is a single long polyprotein and cleaved by the viral proteases into structural and non-structural proteins, the single stranded RNA, which will be translated into a polyprotein. So when we looked at this particular virus, the host range, so it is going to the sugarcane, and the natural host is sugarcane, and it is going only to the sorghum experimentally, but not to any other plant. So we tested many plants, and we found that only it is going to the sorghum, no other plant is getting infected by this particular virus. So we try to look at the physical properties. So biological and physical I'm showing here. So we found that it is showing pinwheel inclusions and laminated aggregates, which is the characteristic feature of Potiviridae family. So with this, we come to a conclusion that this is a member of family Potiviridae, but we really don't know whether it is a potivirus or whether it is a new virus. We don't know at that particular time. So we standardize the purification, and that is the picture that I've showed you just now. And then the electron microscopy picture is showing that again flagellous filamentous particles. One more characteristic feature of Potiviridae, that it is 890 to 15 nanometer. And so then we looked at the code protein. It is single code protein, and then we looked at the amino acid sequence of code protein by doing the tryptic digestion. And we thought that before going into the sequencing of nucleic acid, we will come to an idea whether it belongs to a genus Potivirus, but to our surprise, none of the peptides are matching with the the genus Potivirus members. So this could not give any idea. And then we try to test by getting the antisera from different parts of the India, and it is matching with the virus, which is again uncharacterized from the UP and UK and Parbhani area in India. So they don't know which virus it is causing, but they produce the antibodies. So this virus is going and matching with those viruses antisera, but not with any of the ACMV strains. So with this, we come to a conclusion that this may not belongs to the ACMV as we thought initially. So so now the idea is that it belongs to family Potiviridae, but it is not related to the potivirus that is generally thought as a sugarcane mosaic virus. And actual taxonomic status we will get only through the genome characterization. So you see what I've showed you before, the whole criteria. So we have been using all these criteria one by one, and we come to a last criteria where we get into the genome characteristics. So you see the RNA, which is 10 kb here, and we try to sequence the whole genome. So you can see the overlap strategy that we have used to clone the complete genome, and then we sequenced the complete genome and we got the sequence, and then we try to match with other viruses, and we found that so this particular viral sequence is matching with the Triticum mosaic virus, which has been considered as Poaceae virus member. And that's how we got an idea now that this virus also belongs to Poaceae virus, not the potivirus, which we initially thought about it. So I really want to show you this case study because of the reason that so the characteristic that has been used from the biological, physical, the serological, and the molecular characteristics that we have been used, and for this particular virus, only the molecular characterization, the full-length genome sequence. See, some of the viruses, generally the members of the potty virus, you can even detect the virus. I mean, you can identify the virus based on the partial sequence also, like coat protein and 3' UT are the important regions for the characterization of potty viruses. But still, this particular virus, we could not get the idea by coat protein and 3' UTR sequence, because it has got only not even 10% match we have seen in those areas. So within the genus, if at all, if you want to get an idea, then you have to probably you have to sequence complete genome. Then only you will be see now also this virus is having only 50% homology with the triticum mosaic virus in the whole genome, and only 20 to 30% homology with the rhinoviruses and trichomoviruses. So it is still a lot of disparities there, but that much is enough to put into the genera. So we have, I mean, ICTV has accepted SESMB as a member of the poaceae virus. So this is one thing I would like to show, and then I'm getting into the second topic of the talk, that is diagnosis and detection. So I don't want to go into the details. So diagnosis and detection is very, very important to control the virus diseases. So mere detection of the virus doesn't imply that to the identification of causal agent. Just now, Dr. Amit said that. So most of the times diagnosis is getting into the taxonomic status of that particular virus is very, very important to get into the control strategies. So then why we need to do the diagnosis or detection for the effective management strategies, you need to have those techniques. And for the plant quarantine also, you need to have a accurate and fast, reliable diagnostic techniques, and the nuclear stock production programs. So you should definitely have these things. And then some of the sensitive tests are very, very important for the epidemiological studies. And of course, for the surveillance and monitoring of plant viruses, you need to have these technologies. And so situations where you need to have these technologies, again, I've shown this detection of mixed infections and plant virus interactions, virus vector interactions, and monitoring of viriferous vectors, identification of alternative and collateral hosts, and the sources of virus, whether it is present in water, soil, and crop debris, some of the viruses, especially tobacco viruses, and certification of planting materials, plant quarantine, just now I said, and production of transgenic plants. So in all these cases, you may need to have a reliable and fast, cheap and large-scale indexing test is needed, which is very, very important. Okay, so what are all the different things which will matter for the test is that host plant nature and the concentration and distribution of the virus in the plant. So environmental conditions, mostly temperature and plant nutrition, plant age. So all this matters when you consider the concentration. So what are all the samples that you need to take for the detection? So you may have to take, most of the times people will rely on leaves of different ages. But it is very important to consider the age of the plant when you take the leaf. And then some cases it is bulbs and corms, and then tubers, roots and suckers, stems, seeds, pollen. It depends on the particular virus that and the host plant. You need to decide wherever the virus is present in highest concentration, you may have to consider that part. And in some cases, tissue sections and viral vectors, of course, and the water and the soil, dried plant debris, alternate and qualitative hosts, depending on your experiment, depending on the need that you're going to go for, you may have to decide on that. So again, here also, like characterization, virus characterization, here also you have to consider some of the properties of the virus, where biological activities of virus, where you can go for infectivity assays, and you can go for a transmission to the indicator hosts and light microscopy of inclusions. But again, you know that these activities are little cumbersome and time taking, and it may not give all the time the conclusive information about the identity of the virus. And physical properties, of course, electron microscopy you can do and get the idea about the shape of the virus. And the viral proteins, so you can use some of the antigens like purified whole variants you can use, or isolated proteins. coat protein you can use, or some of the recombinant viral proteins also you can use, and antibodies, of course. If you have a pure protein, then you can raise the antibodies, pure coat protein, polyclonal antibodies, and so you can go for monoclonal and recombinant antibodies also, but they're all a little expensive when compared to. And then in vitro serological tests, if you have a good antibody, then you can go for all these in vitro serological tests. I'm not going into details of any of these techniques. So precipitin tests, latest agglutination, all are conventional and labeled antibody-based tests, and fluorescent microscopy, radioimmunoassay, ELISA, of course, it is a wonderful technique. Still, it is holding very good with respect to the plant virus detection. And in ELISA, you have lot of different ELISAs are there. So every alternate day, you will see one paper on ELISA, different types of ELISA for the plant virus detection. So DAS and DAC and indirect and direct ELISAs, and where you have TAS ELISA, DOT ELISA, tissue blot ELISA, and ISEM also an important technique with respect to the serology. And then virus nucleic acids, again, here you have a list, a lot of different tests are there. So if it is an RNA virus, you can go for DS RNA and DS DNA analysis also with respect to the viral viruses which are having the DNA. And then dot and slot blot hybridization, of course, little less sensitive than the PCR, but still, if you have a good probe, so these techniques are based on the complementarity between the pathogen and the sequence probe that you have. And then polymerase chain reaction, of course, you have lot of different PCRs. So as Dr. Amit said, PCR is very good and reliable technique for the phytoplasma detection. Here also, it is the best method for the virus detection as well. So lot of different PCRs are there, where direct binding PCR, immunocapture PCR, and real-time PCR, and reverse transcription PCR. Multiplex PCRs are there, where two or more pathogens can be detected. And the LAMP, of course, loop mediated isothermal amplification is also has come into the picture now. A lot of plant viruses has been detected using this LAMP method, where you don't need the thermal conditions, denaturation and renaturation of DNA is not needed. And there are some technical advancements. Recently, it has come, I mean, it is not very recent, but a lot of studies are there with respect to plant virus detection, especially the viruses which are infecting the woody plants and the perennials. So some of the microarrays, next generation sequencing has come into the picture, spectroscopic and imaging techniques, and profiling of plant volatile organic compounds, biosensors has come into the picture. Now the IoT, where you can use the mobile app with the help of artificial intelligence. And recently in cassava viruses has been identified very well in Africa, and there is one best example. So I've given a list of different detection technologies which has been used here. So based on biological properties, physical properties, and protein based technologies. So the techniques directly I've given the list here. So symptomatology, bioassay, electron microscopy and light microscopy with respect to inclusions, antibody based tests, in that you have a list again. So lateral flow immunoassay is again picking up because it is a point of care test, where at the field level you can use at the bedside, and it is very, very important technique which has been picking up. And the nucleic acid tests, again you have a list of PCRs and isothermal amplifications. You have a lot of different isothermal amplification techniques are picking up. And then NGS is also picking up, and this is something plant DX and Mini-on technique also is in nanopore sequencing also is a new technique which has been picked up and has been used for some of the plant viruses. And in the biosensors, again, different types of biosensors has been detected based on the protein or nucleic acid of the virus. So what are all the different parameters that you need to consider while considering the detection tests? So you need to see the availability of reagents, machinery, and the sensitivity of the level of sensitivity and specificity that is required. Reproducibility is very, very important feature. Type and number of samples to be tested you need to consider while selecting a test. And expertise and skill is very, very important. So whether you need to have that training or not, you need to decide when you have this the technique that you are going to select for. So what are all the merits and demerits with respect to different detection tests? So what are all the relative merits that you need to consider? Of course, if the technique is sensitive enough, then it can detect the lowest amount of a virus that is present in the plant. So accuracy and reproducibility is very, very important. So if you need to get the consistent results, number of samples, if one operator can detect he can operate more number of samples within less time, that is very, very good and important. Cost and sophistication of the apparatus also very important. If you want to do every virus detection through microarray, it is really, really expensive and it is very costly, so it's not affordable. So the degree of operator training required is also important. So everybody cannot do the microarray, so only the everybody cannot do NGS for that matter. So adaptability to simple laboratories and field conditions. So point of care testing, detection tests are very, very important. So if you have a simple test which can work on the field side, where a farmer can easily use it, then that is the best test. And it should be reliable and cheap also at the same time. So especially for the developing countries like our country, it is very much needed. And then so the biological criteria, so much of problems that you have here, the asymptomatic plants, especially masked and latent plants, it is tough to decide on that. And what symptoms, severe and mild symptoms, sometimes it is confusing. And diagnostic symptoms, some of the symptoms we can detect through the diagnostic symptoms like inhalations in the case of tobacco leaf curl virus. And dissimilar viruses can induce similar symptoms in the given host. That is why it is very confusing. And different symptoms in a given host in the same virus, it can give a different type of symptoms. So ELISA, of course, as I said, it is widely used. It is useful for the large scale indexing, and it can be done in just within 6 to 24 hours. And then crude leaf extracts can be used, and quantitative measurements also can be done. And low cost and long shelf life, simple equipment is sufficient, and economical and efficient use of anti-sera is possible. And then automation and commercial production of kits also possible, and it has been practiced. So apart from that, the optimization of each and every test is very, very important. The removal of inhibitory substances also very, very important. And if you have the inhibitory substances, it may affect the sensitivity. So for evaluation and for minimal amount of purified target virus nucleic acids, we can use for the standardization. And so the variety of laboratory assays for plant virus detections are available, to just conclude, and they all differ in sensitivity, specificity, and appropriateness of use. And there are many new tools and technologies are coming up, and costs and labor needs are significant factors in selection. All techniques and assays may need validation before use for routine testing. And just as a case study, I would like to show you that how we have used the sugarcane streak mosaic virus to use the biochemical, the biological, serological, and molecular tests, how we developed. So here we used DAC and DAS ELISA and bioassay, and we compared, and we found that DAS ELISA is very sensitive for the detection of sugarcane streak mosaic virus. So we developed slot blot hybridization by using both radioactive label probes and dig oxygen in probes. And we used both the purified virus and crude sap for the slot blot hybridization. And some of the limitations that we found with respect to the polyclonal antibodies, that they give some kind of cross reactivity. So we raised the polyclonal antibodies for the recombinant code protein, which has been expressed in E. coli. So we took the code protein gene and we put it into the bacterial expression vector, and we expressed in E. coli. So we took the recombinant code protein and raised the antibodies, and we found that these antibodies are very good and having high titer. And we used this recombinant antibody for the development of DBIA, dot blot immunoassay. And then we standardized the technique, that is ICRT-PCR, immunocapture reverse transcription PCR, and we used for DB-PCR also. And we found that ICRT-PCR is very sensitive when compared to all these technologies. ICRT-PCR, we could able to detect the lowest amount of virus. And we developed a duplex PCR, both ACMV and ACSMV we could able to diagnose in one test. And we could able to detect 3 viruses also, like maize stripe virus and ACSMV and ISMV in multiplex PCR. And some of the potyviruses which are infecting the banana, we could able to develop that multiplex PCR. And this is aeroid viruses, we developed multiplex PCRs. So with this, so all these figures were taken from these publications of our older publications. And I would like to acknowledge all the students who has contributed for this. And so I would like to remember my professor P. Srinivaslu at this point, who is no more now, and who is responsible for most of these studies. And I would like to thank Professor Savitri from Department of Biochemistry, IISc, who has helped me to do most of these studies. And I would like to acknowledge CSR and DBT for the funding for all these studies. Thank you once again for the opportunity, and thank you for your attention.
Thank you, Dr. Hema. Can you close this screen? Your,
yes, sir.
PPT.
Yes, sir.
Yeah.
Yes, sir.
Yeah, thanks for a wonderful, very comprehensive talk on the diseases, especially the sugarcane streak mosaic virus, which was done by your group. I think you and Dr. Savitri came up with a few findings 2 decades back, I think so. It's very wonderful to listen to your talk, very comprehensive talk. I think there are many students in this group. I think they would have enjoyed your talk on the virus diagnosis in depth. We have given the talk. We can take 1 or 2 questions, very quick questions. Any questions, please? Any questions?
Sir, I'm
Okay.
sorry if I've gone too fast, because I know
I
that
understand. Yeah, I understand.
Yeah.
It's very fast, but since the content is very high, so you had to go such a fast. Madam, I would like to ask just 1 question for the benefit of other audience.
Yes, sir.
You said that sugarcane streak mosaic virus and sugarcane mosaic virus, both are one is poaceae virus and potyvirus.
Yes,
And as such, physically the viruses are looks very same. How do you while purifying, how do you differentiate?
Sir, while purification you cannot differentiate, sir. Only through PCR you can differentiate, sir. Only by using the specific primers only you can. So that's why we developed a duplex PCR where we can get both the bands.
So is the PCR is the only best way?
Yeah, only PCR, sir. ELISA also you can do, but ELISA sometimes you get false positives and negatives. So
very good.
PCR is the best method.
Yeah, that's correct. Yeah, that's just for the sake of students I asked this question. So if there are no other questions, I think we can go for the third speaker, because we have got a very limited time, because there are many oral presentations are there. So once again, I thank Dr. Hema, and now I invite Dr. Amrutavalli. She is the Associate Professor in Department of Botany and Microbiology, Acharya Nagarjuna University, and she is one of the very senior person, and working for long time on this particular aspects of microbiology. And she has visited many countries, and she guided more than 15 PhD students, and also she handled 5 projects, 3 as a principal co-investigator and 2 as a co-principal investigator, backed the projects from the DST, UGC, and DST, FIST, etc. And she has published almost 80 reviewed 80 publications in highly rated journals, and wrote 3 book chapters. And all we know here that she recently what is that organized one important PGPR conference, Asian PGPR conference recently last February. That was a wonderful. In fact, I could see a lot of things on that particular conference, Asian PGPR conference at Guntur. And she received many awards, but I'm not going to list those things. And she conducted 3 conferences. And she's also member of editorial board in many journals. I think Dr. Bilal, Padar, can you can mute your mic. some sound is coming. So now I request Madam to present in brief. Maybe you can take 20 minutes, 25 minutes. If you are quickly finishing, it will be useful for other people also. Thank you, Madam. Please.
Thank you. Thank you. Thank you, Dr. Selvarajan Ramaswamy. Am I audible, sir?
Yeah, very much. Please go ahead.
Thank you very much for your fine recollection of our Asian PGPR conference. So, if given the chance again, we are going to have an international next couple of years. Once again, I am very much thankful to the organizers, Dr. Srinivasa Garu and Seshakiran, for giving me the opportunity to share my views with the enlightened group of phytopathology as well as the students. Since I have been working with endophytic microorganisms for the last 2 decades, I have chosen the topic as recent advances in isolation identification of multipotential endophytic microorganisms showing antagonism against fungal phytopathogens. For me here to face this kind of symposium because of the university, Acharya Nagarjuna University, and the great funds given by DST and UGC for doing the research in this particular aspect. Here, already the previous speakers, Dr. Amit Yadav and Professor Hema has given 2 different aspects. The one is a phytoplasma, a pathogen. Second one is a big pathogen with a very small size virus. Now I am going to flip the coin into the second side, how to control these pathogens. What are the methods and means to undergo for controlling the pathogens, for antagonism activities of the pathogens. Here, so
yeah, okay.
Yeah. So the buzzword of the century, this is the year of plants. This year 21 is the year of plants, global year of plants. The buzzword of the century is sustainable agriculture. Now we come to a state where from agriculture to sustainable agriculture, how to maintain the sustenance of the agriculture for future generations. The reasons we are all well known that urban fringe develop increased population, environmental sustainable. Is there any? Am I audible, sir? Am I audible?
Very much audible, but some mobile ringtone is coming. Madam, you go ahead, please.
Okay, sir. Okay. So here, when come to the sustainable agriculture for future generations, there are the 4 main criteria, as we already know, because it is addressing most of the students who are and budding scientists of the nation. The first aspect is greater productivity of agricultural crops. The second aspect is environmental sustainable development, that the productivity should not hinder, it should not become a giant robot to kill the environmental equilibrium, environmental harmony. The third one is acquisition and management of new biotechnologies to meet the demand of population's food, and agriculture and agri-food sectors by bringing the laboratory techniques to the land. A translational research is a need of the hour for sustainable agriculture and to give a handful of food to the future generations. With this aspect, being the microbiologist, I can say the microbes, which are like an untamed area of, for another 2 centuries, it is going to be an untamed area because their diversity is extensive and their activities are vulnerable and they are highly sensitive. They have a double-edged knife. They can cause diseases, at the same time they can cure diseases. So the torch bearers of the sustainable agriculture is the microorganisms. What is the role of microorganism in agriculture is in all directions, in 4 directions. The first direction is plant growth promotion to reach the productivity maximum, and biocontrol to how biocontrol for giving an answer to the chemical inputs, loads of pesticides and loads of synthetic chemicals for curing these pathogens. Biocontrol is another direction which is handed over by the microbes to give some answer to agriculture and horticulture sectors. The third one is the biostimulation. So it is nothing but the immunity, development of immunity inside the plant. The microbes have wonderful features. Some of the features of microbes, they can stimulate the plant to face the disease, not to invade inside. This aspect is a latest aspect in the field of plant microbe interaction. So the third direction of microbial role is biostimulation, and the fourth role is biopesticide. Here I have separated the biopesticide from biocontrol because biopesticide is involved with another group of living organisms, insects. So here there should be a harmony between the entomologist and microbiologist as well as the agriculturist. That is why we cannot consider the biopesticides as the biocontrol agents. Biopesticide is a different aspect when come to the field of agriculture and horticulture to meet the demand of food production and food sector development. So the torchbearer of the present topic of mine is basically on biocontrol, how to control fungal phytopathogens which are causing diseases in fruits and vegetables. So the main aspect of my research is on fruit and vegetable health. The torchbearer of this biocontrol, it can be answered by all four major types of microorganisms in microbial biosphere: bacteria, fungi, cyanobacteria, and mycorrhiza. All these kind of microorganisms can give an answer for controlling phytopathogens. So these biocontrol agents, the bacteria and fungi, how to select. The main aim of my topic is to give some information to the youngsters how to select a microorganism, how to isolate a microorganism, how to screen that microorganism and prove it as a potential antagonistic against phytopathogens. That the fundamental need is the habitat where from we are collecting these microorganisms. Are we collecting it from the soil, or from the rhizosphere, or from the epiphyte, or from phylloplane, or from rhizoplane? So I think the August fraternity here, they all understand what are these terms and the organisms which are present surrounding the N in number and their mode of action, how they are going to control. Like the human beings, we have polio vaccine for prophylactic, and we have paracetamol as a therapeutic. Similarly, the biocontrol microorganisms which are used, they have both the activities. One is the therapeutic activity as well as the prophylaxis. So how do these prophylaxis? Here come my field of study is endophytes. Here I am going to give the little information what I have with my endophytes. How these endophytes mode of action as a therapeutic agent or prophylactic agent against common fungal phytopathogens which cause diseases in fruit and vegetable crops, especially in the Solanaceae members. So they come to the endophyte. Endophyte is an organism which is resides inside the plant. It is present inside the plant. It invades into the plant through root hair. Sometimes it goes into the plant through the stomata. Sometimes it goes into the stem through wounds. Whatever the way of invasion inside the plant, they coexist inside the plant. They distribute throughout the plant right from the primary root to the shoot apex. Their distribution varies from species to species and genera to genera. In our laboratory, we have done the isolation of endophytes, nearly about 160 endophytic bacteria from chili plant, and their distribution and their population variation in different parts of the plant. These endophytes, they exist inside the plant either intracellular or intercellular Sometimes they go along with the sap in the xylem, and sometimes they will be stationary inside the phloem, and their coexistence, it's a wonderful commensalism, and they will take some minimal nutrients from the plant and elicit some of the wonderful and beautiful secondary metabolites which give immunity to the plant. That is why these endophytes are the novel area of research for the development of biocontrol agents. So with this idea, we have chosen the endophytes as one of our targets to give an answer to biocontrol. So these endophytes, they can be colonized simultaneously by large variety of endophytic bacteria. A single plant can have n number of species of endophytes. A single endophytic bacteria can be seen in n number of higher plants. Endophytic bacteria colonize an ecological niche similar to that of phytopathogens suitable as biocontrol agent. So this is a wonderful word. So the endophyte and the phytopathogen both are having the same ecological niche. A good man and a bad man. A good man always never interfere with the environment. A bad man always interferes with the environment. Here, we are cutting the diamond with another diamond. How to control this phytopathogen, which is present in the same ecological niche of the biocontrol agent, is one of the recent aspects of research in agriculture. Endophytes spans a significant range in both. These endophytes, they are both gram positive as well as gram negative. Since the time is very limited, I am not using the animation. So these endophytic mechanism, the endophyte, as you all know, that endophytes have two different kinds of mechanisms, like rhizosphere bacteria, like mycorrhiza, and all other chemical things. So this is a direct metabolic mechanism is for promote plant growth, and the indirect mechanism is the biological control. This is the basic knowledge one should acquire before going to do some research on endophytes as biocontrol agents or plant growth promoting bacteria. So plant growth promotion mechanism is by attributes like auxins, cytokinins, and gibberellins. Like higher plants, these endophytes also have the ability to synthesize auxins, cytokinins, and gibberellins. They have a wonderful property of phosphate solubilization. With my 2 decades of research, we came to a conclusion that the phosphate solubilization ability of endophytes is lesser than the phosphate solubilization ability of rhizosporic bacteria. Why? Because they are present inside a wonderful 5-star host. They need not have solubilization. They can get a readily available organic phosphates, ATP, to the survival. So this is one of the observations we found in our research laboratory. However, the phosphate solubilization ability of the endophyte also gives a good response for plant growth promotion of the host. Ammonia production and nitrogen fixation, the ACC deaminase. These are the 5 important aspects which have the direct mechanism of endophyte to act as a good plant growth promoter. When come to the indirect mechanism, these endophytes have the ability to produce secondary metabolites which are having the antibiotic property. Antibiotic production, they have the cedrophore production for reduction of iron availability, HCN production, chitinase, protease, and lipase enzymes, lytic enzymes. The chitinase enzyme, existence of chitinase enzyme of endophyte is a wonderful answer for controlling fungal pathogens, because fungal mycelia is made up of chitin. The endophyte, which is having the chitinase, they can dissociate the chitin. They can the wall of the fungal spore. So they may similarly the proteases and lipases. And the ultimate thing is the induction of systemic resistance. So these are the biological control mechanisms exhibited by endophytic mechanism. Unless and otherwise we prove the endophyte what we selected is having these properties, we cannot proceed further for developing these endophyte as a biocontrol agent. So how to do this? How to identify these endophyte is having these plant growth promoting traits? How do they control phytopathogens? How do they produce antibiotics? How can we? There are quantitative and qualitative estimations are the preliminary steps to identify an endophyte as a biocontrol agent. So this endophytic microbial biological control, endophytic is, you have two types, therapeutic and prophylactic. In the therapeutic, because of their hyperparasitism nature and antibiosis, they can control the fungal pathogens. When it comes to the prophylactic, without having the fungal pathogen inside, the inoculation of the endophytic microbial biological control, biological material, or application of these endophytic bioformulations to the plant, they can develop a systemic acquired resistance, or they can induce a systemic resistance by enhancing the ROIC system inside the plant. So I need not explain all the things because majority of the audience here, they know that. So some of the ISR ROIS system are peroxidases, polyphenol oxidase, phenyl ammonia lyase. These are some of the enzymes which are reactive oxygen systems. They enhance, if once they are enhanced inside the plant is highly immune against any sort of fungal pathogen. Otherwise, the systemic acquired resistance, the existence of salicylic acid and jasmonic acid, ethanol production will be induced by endophytic application. This is called prophylaxis, like our polio vaccination. This is the one way of application, and this is another way of application. These are the mechanisms they speak about the control of phytopathogens. So comes to here, these endophytes, there are two different aspects. How to isolate? I want to emphasize more on this part because we have done these experiments since 20 years. So the isolation of endophytic microorganisms, it has regular steps like any other microorganism, any other bacterial or fungal microorganisms we isolate in a microbiology laboratory. The preparation of media, here one should be very careful. If it is endophytic bacteria, you need to go for NAM. If it is a fungi, you need to have more precaution. The endophytic fungi, majority of the endophytic fungi are sensitive to dextrose. The fungal medium which is having dextrose may not be suitable for isolation of this endophytic microorganisms. So the preparation of medium is one of the critical area for isolation of the endophytic microorganisms. Like virus, as told by Professor Haima, any part of the plant can be used for isolation of the endophytic microorganisms. Being a biological scientist, I always prefer to isolate the organisms from parts of the plant which is not economically useful to the human beings. In our laboratory, we have isolated endophytic microorganisms from the root of the chili, fruit of the chili, stem, and green fruit, red fruit of the chili. We have also done the experiment with isolation of fungi with Ecorus calamus, Vasakumu antar. It is in English, it is called sweet flag. We have isolated wonderful 4 fungi from Ecorus calamus, which act as a wonderful biocontrol agents against Coltotrichum. 4 different species of Coltotrichum cause anthracnose in chili. So the preparation of medium is one of the crucial steps. Then comes to the serial dilution. As you know that if it is a bacterial dilution, at the 10 power of 6 dilution, we can get the bacteria. At the 10 power of 5 dilution, we can get the fungi. Then comes to the surface sterilization of the plant material. In the next slide, I will emphasize how this is done. This is a very important step in isolation of the endophytic fungi. Apart from that, the confirmation of endophytic fungi. How can you confirm this is an endophyte? It is not an organism which is present on the plane of the root. It is not the organism which is present on the plane of the leaf. These 2 steps are very much essential for avoiding the adulteration of the normal rhizosporic bacteria with endophytic bacteria or fungi. This is one aspect. The second one is selection of endophytic microbial isolates showing fungal antagonism. How to do the screening of fungal antagonism? The fungal antagonism screening can be done in 2 ways. One is a qualitative, another one is a quantitative. The qualitative antagonism can be done by dual culture method. I will show in my coming slides. And the quantitative estimations of siderophores, quantitative estimation of volatile compounds, quantitative estimation of antibiotics by using GCMS analysis, LCMS analysis. So that is a high end of research. Before going to the high end of advanced research, one should have the quality, one should have assure the quality in the first stage of selection of an endophyte as a fungal antagonistic organism. Before spending lakhs and lakhs of rupees for identifying an organism having the particular gene, the screening, which is the fundamental screening of fungal antagonism with perfection, like we have the good fundamentals in our earlier stages leads to the good fruits at the later stages. We can reap like that. So these two aspects I'm going to enlighten here with my limited knowledge. Comes to the isolation, the plant materials isolated, the plant material what we have shown, we can select any part of the plant for isolation of the endophyte. The first step is they have to be disinfected with H2O2 for 2 minutes. Why it is only for 2 minutes? For bacteria or fungi, it is we have standardized it for only for 2 minutes. Why it is more than 2 minutes? It is a question. If a budding researcher, if he wanted to disinfect it more than 2 minutes, what will happen? Here a little bit of chemistry is needed for the scientists or researchers. Why it is only for 2 minutes? The time period is very important. This is a different topic that we can discuss later. So H2O2, the peroxide disinfection for 2 minutes, followed by rinsing with 70% ethanol for 5 minutes. What will happen if we have rinsed it more than 5 minutes? It's an alcohol. It is going to dehydrate the plant material. If once it dehydrates the plant material, there is a desiccation to the organism, endophytic organism present inside the tissue. So that is why here the 5 minutes we have standardized in our laboratory for the endophytes from the specific host. This is the 5 minutes. Followed by 3% sodium hypochlorite rinse for 10 minutes. We all know that sodium hypochlorite is the disinfectant we are using since March 23rd, 2019 onwards till date for washing our hands and for cleaning our. This is a best antiviral and antibacterial solution. 3% sodium hypochlorite rinse for 10 times, followed by a rinse in sterile saline water for 5 minutes each. So 5 minutes each, saline water with. Then comes to the sterility checks carried out as per the procedure of, this is a wonderful paper where he has given a clear picture of how to do the sterility check. For endophyte, for confirmation of any endophyte, this sterility check is very, very important. You have to check the microorganisms in each and every step of wash. In the wash of a organism. So in the sterility checkup was done by imprinting the surface sterilized plant tissue on nutrient media for culturing aliquots of water from the last rinsing onto nutrient medium. Here, clearly I have given a statement, last rinsing onto nutrient medium in order to confirm the surface disinfection and verification of surface free from biological contamination. Till the surface of the plant material what we have chosen is not giving any microorganism, up to that we have to wash with saline water to particular plant tissue or whatever the part of the plant what we have chosen for isolation of the endophytes. This sterility checkup is very essential. Then come to the selection. So this is the article, this is from internet I got because it has wonderful pictures. So the common fungal pathogens, phytopathogens, the fungal pathogens, common fungal pathogens which cause diseases in fruits and vegetables are Fusarium, Rhizoctonia, Bipolaris, Botryospirata, Alternaria, Coltotrichum. These are all the common, I have taken this from this article. Here, these are the endophytic fungi. These are the endophytic fungi which have shown inhibition of the particle. These are the fungal phytopathogens. These are the endophytic microorganisms. These microorganisms are showing the reduction in the size of the growth of phytopathogen in a jewel culture method on PDA medium 4 days after incubation. Here a question arises. Is it because of competition? Is it because of hyperparasitism? or is it because of antibiosis? Simply by having the reduction in the size of the mycelial growth, fungal mycelial growth, we cannot conclude that this endophyte is having the antagonistic property. It is one of the aspects to screen. It is having some antagonism. It is showing some mechanism to control the fungal phytopathogen. Here we can see the total radial growth has been reduced here. When come to here, the radial growth, the growth is more longitudinally, and the place of endophytes, the growth is reduced. Here, it is dominating the growth. The growth of the pathogen is dominating over the endophytes. The same endophyte speaks in different languages with different phytopathogens. This is an aspect to be answered. This is one of the future answer. Is it hyperparasitism, antagonism, or competition? They come to this is in our laboratory. We have done with 4 different types of Coltotrichum species, isolated from different varieties of chili, and identified them as different species in 16S rRNA partial gene sequenciation. This is a Coltotrichum, and these are the endophytic fungi isolated from the red fruit of chili. They have, in dual culture method, they control the Coltotrichum. This is one of the example I have shown. I am very happy to share with you all. We have 76 endophytic bacteria and 12 endophytic fungi which have potential control over the Coltotrichum species, and we have one patent on this also. Come to this. These are some of the endophytic fungi which have completely, this is another test. This is antibiosis test. Here, the dual culture method, one plate is with endophytic microorganism, another plate is a fungal pathogen. The volatile compounds which are emitted by these endophytic organisms reduce the growth of fungal phytopathogens. It clearly enmarked that it's some volatile compounds isolated, liberated from these endophytes are having antibiosis against these fungal pathogens. So this gives the second confirmation it can be a future potential biocontrol agent. These are the volatile substances liberated. So means some of the volatile compounds which are liberated from endophytes are having biocontrol potentially against fungal pathogens. Comes to the third aspect. When we specifically speak about the fungal phytopathogens, the fungal cell wall is made up of chitin and cellulose. So these are the simple basic tests needed before going to the advanced technology. So with minimum amenities, how to get the maximum information, how to expect a maximum properties, potential of the microorganism is the need of the all biology students and all biology researchers. So this is a simple test. By using the CMC agar plates, carboxymethyl cellulose carbox plates. So this is one of the cellulose activity test, protease activity test by skim milk agar medium, and glucanase activity test by using Pachyma and solid medium supplant. So this type of confirmation, after confirming in a dual culture method, after confirmation with the volatile oil production, then we will go for the lytic enzymes, endophytic lytic enzymes which are present there with the specific fungal organisms. This gives an idea. Yes, it is having both qualitatively, it can be qualified qualitatively, this organism is qualified to act as a biocontrol agent. Before going to my suggestion, before going for further proceed of identification of the biocontrol agent by using lots of revenue on 16S rRNA and GCMS, it is a simple test. This test I learned from Indian Institute of Horticulture Research in the laboratory of Dr. P. Chaudappa. This is a leaf aggressiveness test. This, I think, being the agricultural scientist and horticulture scientist, you all know this. This is a simple test. It gives an idea whether it suppresses the aggressiveness or not. So this is a treated plant, and this is endophytic treated leaf, and this is a untreated control plant with both are treated, both are given with fungal phytopathogens, and this leaf is given with endophytic culture incision, endophytic incision. After 14 days, we could able to identify that the endophytic inoculum treated incision leaves are healthy and they have not shown any symptom. This is another preliminary screening test. Though it is a preliminary screening test, it gives 50% of solution to our problem. We can confirm that this is a potential biocontrol agent. Then,
Madam, sorry for interrupting. Already
okay,
30 minutes are over.
I will finish it up, sir. For a few more slides are there. So
Yeah.
the inoculum, the treated with fungal pathogens, the gradual from A, B, C, D, E, F, G, gradually the mycelial growth is reduced. This is the electron microscopic photography also has given an idea that these organisms are reducing the growth. In a pot culture, the co-inoculation of phytopathogen with the endophyte, we can see that biological suppression of Coltotrichum gliosporides in our laboratory. It's a challenged plants are grown very weak than that of the treated plants. This is another confirmation. After that, the morphological characterization of the endophytes, which is a routine procedure. Here, from our case study, from our laboratory, we have about root endophytes of 10 root endophytes. We identified them with it is a regular procedure of morphological characterization, and then followed by biochemical characterization with oxidase, nitrate, reductase. These are the biochemical characterization. Is a fundamental test for identification of these organisms. Here CR8 has shown positive to some of the enzymes. So CR8 and CR20, the blue color, red color isolates are going to be much useful when compared to the all 10. This is how the screen, the biochemical characterization. Then come to the microscopic observation. After biochemical physiological micro observation, we came to know that the 10 isolates we have isolated from the root of chili, they have Enterobacter, 2 Enterobacter species, 2 Klebsiella, 1 Rhizobium, 2 Agrobacterium, 2 Enterobacter, 1 Klebsiella. So all these 3 Klebsiella are not one and same. Here, the advanced technology of molecular identification is needed. These 2 Enterobacteria, do they are one and same or 2 different? Here, we need to have the molecular identification. So this is a total picture how to separate one organism with the other organism out of the colony by colony morphology. So these four different types of organisms are categorized. By biochemical and physiological, we tentatively identified 6 different genera here. By morphological, by 16S rRNA, as Amit and Hema told, 16S rRNA and 18S rRNA genetic material. Here we, by 16S rRNA molecular diagnosis, clearly give these Enterobacter and these Enterobacter are not one and same. Phylogenetically, these 2 are different. And these Klebsiella and these 2 Klebsiella are phylogenetically different. These 2 Agrobacterium are different. So the morphological identification has gives each strain is strain specific, and we have deposited all these strains in NCBI with specific accession numbers. This is how the 16S rRNA gene sequence is helpful for diagnosis, for identification of a potential antagonistic biocontrol organism. So these functional attributes of this, by functional attributes, 106 out of 106 CER endophytes from the root, only 1 to 80, 28 have shown the antagonism, and 10 isolates shows multiple plant growth promoting activities as well as the antagonism. These are considered as the multiple endophytic microorganisms. So come to the last stage. This is called prophylactic. In the prophylaxis, this is the blue colored dots shows that these are the natural immunity in a healthy plant. When we apply biocontrol agent to a healthy plant, it is ready with elicited proteins that is called jasmonides, jasmonic acid, ethanol, etc. and etc. to the lesser record. When you are given with a challenged plant. When you apply this biocontrol agent to a challenged plant, the challenged plant mode of action is different from that of the therapeutic action. So this is more useful than this giving the treatment to a challenged plant, giving the treatment to a diseased plant. Instead of that, by seed priming, if we are able to incorporate biocontrol agents into the seed in the nursery beds itself, it develops an induced systemic resistance inside the plant, so that it acquires a natural immunity to fight against future invading microorganisms. So the invaded induced systemic resistance can be confirmed by 3 different enzymes: peroxidase and polyphenol oxidase and phenylammonium enzymes. So the take-home message from my talk is, for the researchers and budding scientists, so aspects to be considered for evaluation of the endophytes as a biocontrol agents. The aspects what we have to be considered is the antibiosis, induction of plant resistance, interference with the virulence of a pathogenic target organism, how it grows inside the plant, how many colonies per unit CFU values, how it is root colonization. These are all have to be done by electron microscopic studies, competition with ecological nutrients and habitats. These are the things to be considered when evaluating and assessing a microorganism as a biocontrol agent. My acknowledgements to UGC and Angro, especially for Acharya N.G. Ranga Agricultural University, for their suggestions for in selection of my chili plants and varieties of chili plants for isolation of endophytes. My special thanks to Acharya N.G. Ranga Agricultural University, Lawn. This is the team with which I have worked with endophytes, and all of the fellows who are here, they got their doctorates, and these 3 are working for doctorates right now. Thank you very much.
Thank you.
Thank you very much.
Thank you, madam, for your very elaborate presentation on this endophytes and very interesting talk. And of course, very good contents were there, and it's a really great thing to listen from your laboratory team on the endophytes, endophytic microorganisms, starting from the isolation and till how it actually acts upon on the disease control, pathogen control. We have given very nice talk, and I think it will be very useful for all the students and other young researchers. Thank you very much. And because we are not having time, so we will go for without questions. We will go for, if any questions, you can put it in chat box, and I request Dr. Amruta to answer in the chat box for those questions. So now I think now we'll go for oral presentation. Oral presentation, there are 10 actually. I don't know how many of people are here, but straight away we'll go now. I think I request Dr. Parameswari. Madam, can you close this?
Sir, one of my scholar has oral presentation. Tulsi Bai, she is ready right now. She may present it from my laptop with your permission. Tulsi Bai, she is already submitted.
No, first Parameswari is there, madam.
Sir, so give me. So after that, sir, in a sequence.
No, I think she can wait. She can wait.
Okay.
She can wait. So now I request Parameswari to present
Thank you, sir.
on real-time PCR-based screening for identification of resistance against sugarcane yellow leaf virus in Indian sugarcane germplasm. And the time given is only 7 minutes. Kindly see that you complete 7 minutes, and we can have 2 minutes for discussion, because we are really running short of time. Though I think probably they would have given 10 minutes earlier. Now I am reducing, cutting short to 7 minutes. So be quick and not to give too much of introduction. Straight away you can go for the point what you want to tell. Okay? Parameswari, please, you can share.
Good evening to one and all. Now I am going to present the real-time PCR based screening of Indian germplasm to identify resistant against sugarcane yellow leaf virus. Yellow leaf disease of sugarcane caused by the sugarcane yellow leaf virus. And
share the screen, Parameswari. Parameswari, share the screen. Your PPT, how to share? No? You can share.
Any trouble? Just to open the PPT, keep it open and keep it in your desktop. Then you can share it.
In the Zoom, down you see that green color share screen is there. Then you press on it, and then wherever that file is there, you can start.
Yeah, first you have to open up and keep ready there. Then you can
Yes,
share.
sir, it is open only, but share option it is not showing.
Down, down. It is the share
Down
option
is
is
there,
there.
sir, but share option I have opened, but the desk.
No, maybe the host. No, host has put only. No, he has to allow it for share for multiple participants like that. I think.
Because here I am not able to share.
No, other people are sharing. No problem. Or otherwise you mail me. I will share. Or otherwise you can mail to Selvarajan sir. He will share. He will share the PPT. You mail them. You are mail to both of us.
I think from IPS side they can say whether it is they allowed to share or not.
No, other people are sharing. No, then it, I don't think that is not the problem.
Yes.
Yeah, it is only one participant. I have changed the settings. No problem. Earlier it was one participant can share at a time. Now I changed it to multiple participants.
Okay,
Parameswari, you can share now.
Yeah.
Oh, yes.
Okay.
Now you share.
Yeah.
Is visible, sir?
Yeah, wonderful. Go ahead.
Okay, good evening to one and all. Now I am going to present about the real-time PCR-based screening of Indian sugarcane germplasm to identify resistant against sugarcane yellow leaf virus. Sugarcane yellow leaf disease of sugarcane caused by sugarcane yellow leaf virus, important viral disease, and causing 39 to 43% can growth reduction, and also causing 30 to 33% yield loss in juice. In India, it is first reported by Dr. Viswanathan from SBI Coimbatore. In India, only complete genome sequences are available, but ICR sugarcane breeding Coimbatore is maintaining approximately 600 parental clones, but there is no research is going on in India about the resistance against this disease. So, based on this, we have done real-time PCR to identify the resistant germplasm, followed by the genome-wide association studies to know the resistant mechanism through genome-wide association approach. In National Hybridization ICR Sugarcane Breeding Institute Coimbatore, more than 600 germplasm were maintained. Resistant plants were identified through phenotypic symptom expression and also for the quantitative real-time PCR for the year 2018 and 19. The yellow leaf disease severity grade was calculated based on the 0 to 5 yellow leaf disease rating scale. In case of 0, there is no symptom. In case of 1, midrib yellowing is there. In case of 2, prominent yellowing of the midrib on the leaves and no bunching is observed. 3, progress of midrib yellowing from laminar region to tip towards and bunching of leaves. In case of 4, drying of laminar region from leaf tip downwards along with midrib and bunching of the leaves as tuft. In case of 5, standard growth of the can is occurred, followed by the drying of the whole can. Based on this, 4 is calculated, 0 to 1 is resistant, 1.1 to 2 is moderately resistant, 2.1 to 3 is moderately susceptible, 3.1 to 4.1 to 5 is highly susceptible. Based on this phenotypic screening in National Hybridization Garden, their breeding from the India, all the sugarcane breeders will come there and crossing will take place. So they are maintaining the approximately 600 germplasm. These are the different types of phenotypic symptom expressed notice in the National Hybridization Garden of ICR Sugarcane Breeding Institute, Coimbatore. From there, 150 yellow leaf disease free samples in resistant category and 125 samples under a moderately susceptible and highly susceptible category were collected for the 2 years, 2018 and 19. So based on the apparently symptom-free samples and also from the susceptible samples, real-time PCR was carried out. Further, for genome-wide association approach, single nucleotide polymorphism genotyping were carried out. Here, RNA extracted and cDNA was synthesized, and sugarcane allovirus code protein specific primers were designed, and a positive amplification of 600 pairs, and in case of quantitative PCR, 118 base pairs. This total RNA was isolated from the sugarcane yellow leaf virus free samples, that is apparently free, based on the phenotypic symptom expression. Similarly, total RNA was also isolated from the susceptible samples, that is more than 3 grade of 0 to 5 yellow leaf disease grading scale. Followed, the gradient PCR was carried out. The primers were designed for 615 base pairs primer of code protein gene was used, and these are the results. Followed, so the confirmed samples, again susceptible samples, again quantitative real-time PCR primers were designed based on the consensus of code protein regions of the sugarcane yellow leaf virus. 2 primers were designed. This is the primer set 1. This is the primer set 2. To standardize, the RT-PCR was standardized using these 2 real-time PCR primers. Then in case of first QRT-PCR primers, we didn't get the best results. So next PCR primers has given 181 base pairs, then standardized. These are the RT-PCR based detection of the sugarcane yellow leaf virus resistant and susceptible sample. In case of resistant samples, no amplification was observed using the quantitative real-time PCR primers, and in case of susceptible samples, 181 base pairs that virus was detected. So like that, for 150 resistant samples and 125 resistant samples, we have identified. After identification of this these are the different co-clones. Co refers to Coimbatore, because in Coimbatore only breeding crossing will be carried out, and all the sugarcane breeders will come to Coimbatore and they will cross. So CO Coimbatore, the COH is Haryana. So first name is Co, it will come, then Haryana, Co, OR, Orissa, like that. So only 11 samples, we didn't get any amplification. Those are 70, COSNK 14101, COPB 1182, COLK 97147, COPAN 96219, COBLN 03175, and etc. These qPCR results, this standard curve are optimized using SUILP code protein of CO86010 plasmid. These are the results of the quantitative real-time PCR. These are the gel pictures of the resistant samples. In quantitative polymerase chain reaction assay, that resistant and susceptible samples, total collected resistant samples are 150. In that, positive are 139. 11 are resistant. Susceptible samples, all are susceptible. So out of 150 samples, only 11 samples. This visit 28, it is exotic hybrids. Are truly resistant to the yellow leaf disease. So after this DNA was isolated and given for single nucleotide polymorphism based on the can chip array, 45 can chip array developed by the Australia to find out the SNP resistant markers. Thank you.
Thank you, Dr. Parmeshwari. It's very nice work which you have presented on the resistant source for sugarcane yellow leaf virus, which is very important problem now across the country. So you have come out with some around 11 resistant clones. That's very nice. So since we do not have time, let us not take questions. But again, if there are any questions, kindly you can give it in chat box, and I request Parmeshwari to answer. Okay, at the end we will compile everything. I think our reporter is doing the job of compiling all the things. Now I request Dr. Bharati Bhatt to give a talk on status of sunflower necrosis disease. And 7 minutes are given, so don't take too much time on introduction, and you can go ahead. Dr. Bharti Bhatt. Dr. Bharti Bhatt.
She's there online, but I don't know why.
She's there, yeah, I saw. She's there very much.
Let me call sir. I'll call them.
Yeah.
Or otherwise, you can go to next speaker, sir. She will, by the time she comes
the
back.
next speaker is Dr. Miss Tirumala Raju Chandana.
Yes, sir, I am there.
Okay, are you ready?
Yes, sir, I am
Chandana,
ready.
are you ready? You can talk on studies on pathogens associated with gummy ooze secretion in bottle gourd and their management.
Yes, sir, I am ready.
7 minutes are given for you, so try to finish quickly.
Okay, sir. Sir, did you able to see my screen?
We are seeing your entire what is that? Your desktop. You just pick up. Yeah, I think here is there your presentation.
Sir, now it's visible.
You just
sir?
no, it's not visible. You just
Hello.
click.
Hello, I am Bharti Bhattiar. Shall I share my screen now?
Are you ready, madam?
Yeah, I am ready, sir.
Then okay. Chandana, you wait.
Okay, sir. Okay, sir.
Okay, you can just stop your share screen.
Yes,
Allow Dr. Bharti Bhattu to share.
Okay, sir. Good afternoon, everyone. So first of all, I thank the organizers for giving me this opportunity. So today I am going to present the topic on status of sunflower necrosis disease. So this work was carried out at Hyderabad in the Department of Plant Pathology as well as DOR. So coming to sunflower necrosis disease.
Madam, share your PPT.
Okay. Share చేయాలంటున్నా. Is it visible, sir, now?
Yeah, very much. You can go ahead.
Okay, sir. So it's caused by tobacco streak virus, which belongs to genus Hilar virus and family Bromomyridae. So coming to introduction, it was observed in serious proportion up to 80% in different parts of Karnataka, and later also in Andhra Pradesh, Karnataka, Maharashtra and Tamil Nadu. So it occurred in epidemic form consecutively for continuous 3 years, and incidence ranged from 10 to 80%. And this disease, it's not seed-borne in nature, and it's mainly vector transmitted. And main source of inoculum was identified as pollen grains of the infected plants, especially Parthenium. So these are the different vectors. It's thrips, different species like Cytothrips, Frankliniella, Megalurothrips, and Thrips palmi. And mainly the disease, it is spread through weeds also. These are the different weed species which are found in and around sunflower fields. And especially Parthenium, it plays a major role in the transmission. And coming to symptomatology, these are the characteristic field symptoms, which includes extensive necrosis of the leaf lamina, petiole, as well as stem. And early infection, it kills the plant, as well as severe stunting and malformed heads with chopped seeds. And these are the different symptoms which are observed under the field conditions, like chlorotic spots, then leaf necrosis, bending of stem, stunting of the infected plants, partial grain spilling, then black streak on the stem, necrosis of the black. And these are the symptoms which are observed under artificial inoculated conditions, like mosaic symptoms, chlorotic spots on the leaves, as well as necrosis of the leaves. So coming to host range, around 43 plant species belonging to 8 families were tested for susceptibility to SND under artificial conditions. And observation for symptom expression was taken after 40 days. And the plants expressed localized symptoms like chlorotic local lesions, then necrotic local lesions, mosaic mottling, as well as systemic symptoms like chlorosis, mottling, oak-leaf pattern. And these are the different plant species which are susceptible to sunflower necrosis disease, like different species belonging to different families. And both systemic and localized symptoms were observed on different plant species belonging to Asteraceae, as well as Chenopodiaceae, and also Leguminosae, Malvaceae, and Solanaceae. And these are the different local lesion hosts of sunflower necrosis disease, like Chenopodium, and then Dolichos, Nicotiana species, and Vigna unguiculata. And these are the different symptoms on local lesion hosts. We can see chlorotic local lesions on Chenopodium, and also you can see chlorotic local lesions on Chenopodium quinoa. And these are the symptoms on cowpea variety C152, and also Pusa komal. And you can see chlorotic local lesions on Dolichos lab, as well as necrotic symptoms on macrotilloma, it's a horsecomb. And here we can see beautiful symptoms like white ring-like pattern, as well as oak leaf pattern on tobacco leaves. And coming to detection of this particular virus, so especially serology, so we can identify the virus, and this is the protocol for DAC ELISA. So here we can see the presence of virus by indication of yellow color. And another method is by electron microscopy through leaf dip preparation. So by using TEM, we can see virus particles which are isometric in shape. And coming to epidemiology, weeds play major role in transmission of the disease, especially 35 weed species which are found in and around sunflower fields. So they were tested for the presence of TSV by using assay host, and around 12 weed species they were found infected by the virus. And there are some symptomless carriers also, like Parthenium. And these are the experimental host range by artificial inoculation. So there are some symptomless carriers also, like Amaranthus and Parthenium, which is confirmed by assay host as well as serology. So coming to effect of date of sowing, here we have used a variety modern, so which is sown at monthly intervals, and 2 rows of SND inoculate plants. It's also used as infector rows, which is sown 10 days before. And the incidence of SND as well as population of thrips was recorded. So here you can see effect of staggered sowing, especially during June and July. The thrips population was maximum, so it can be correlated with the incidence of SND. So during Kharif, especially in the months of June and July, you can see higher incidence of sunflower necrosis disease. So coming to screening of different germplasm as well as cultivars and hybrids against sunflower necrosis disease. So here we have tested around 20 R lines and 12 CMS lines, as well as 100 germplasm lines. And observations as were recorded with respect to symptoms as well as % disease incidence. And also we have categorized different lines based on the disease incidence as immune and susceptible. And even disease severity was also recorded in different germplasm. And here none of the varieties were found to be resistant. Only few lines they were moderately resistant, and 69 lines they recorded more than 80%, means they were highly susceptible for sunflower necrosis disease. So here we can see detection of TSV in different germplasm lines by employing DAC ELISA. So most of the lines they were susceptible for the disease, and they were categorized into 7 groups based on the symptoms as well as reaction in ELISA. So coming to management of sunflower necrosis disease, so around 15 treatments were imposed, including cultural practice as well as use of different chemicals as seed treatment as well as spraying, and also intercultivation with red gram. So among these 15 treatments, one treatment that is T11, which comprises of border crop and seed treatment with thiamethoxam as well as 3 sprays of thiamethoxam. So it yielded maximum compared to other treatments. So here we can see the treatment involving intercrop with red gram. So among all the treatments, T11, so it yielded maximum, and also have recorded less population of thrips as well as less disease incidence. And regarding detection of TSV isolate, we have employed RT-PCR method by using different techniques like cDNA synthesis, then PCR amplification as well as gel electrophoresis. And here you can see amplification of bands using specific primers. So all the isolates they were similar. There was no much variation. And regarding this particular aspect, here the virus causing SND, it has a wide host range, infecting many species and also many families. And here the electron microscopy, it revealed presence of typical isometric virus particles, and based on symptomatology, transmission and host range, so it has been identified as the isolate of TSV of ILAR virus group. And out of many weed species found in and around the crop, so Parthenium, it was recorded in a higher frequency, and incidence was higher in July sown crop followed by June sown crop. So these are the different findings of this particular experiment. So coming to screening, so none of the germplasm varieties were resistant to this particular virus. So almost all the lines were susceptible. And in IDM trial, seed treatment with thiamethoxam as well as 3 sprays of thiamethoxam and 3 rows of sorghum as border crop. So it yielded maximum. And regarding detection by using RT-PCR technique, so all the samples, so they were similar. There was no variation. And we have also used infectivity assay by using cowpea cultivar C152 and also Nicotiana. But here also variation was not exhibited. That means so almost all the isolates which were collected all over India, they were similar. And coming to recommendations to the farmers, so we have to avoid monocropping in order to avoid the buildup of inoculum, and also roguing of disease infected plants as well as avoiding cultivation of highly susceptible cultivars. So these are the different recommendations for the farmers. Especially we have to remove Parthenium, which will serve as a reservoir of TSV. And coming to future line of work, so systematic surveys required for the occurrence of sunflower necrosis disease, as well as further screening of germplasm lines, and also molecular characterization of the virus associated with the sunflower necrosis disease in order to identify the strains, and further studies to ascertain the prevalence of TSV pathotypes or serotypes, and also molecular detection of TSV in weeds as well as thrips, because sometimes it may be vector borne. I mean, we should be controlling the vector in order to control the disease. So we have to go for not only disease resistant varieties, also vector, we have to see for vector resistance also. So once again, I thank the organizers for giving me the opportunity, and also I thank our department, PJTSAU, as well as Indian Institute of Oilseed Research for providing me the opportunity to carry out the research work.
Okay.
So thank you one and all.
Thank you, Dr. Bharti Bhatt, for a wonderful presentation on this important disease in Andhra Pradesh, sunflower necrosis disease. And I think again we cannot take questions, but my one question is that can you in chat box you can answer that? That is
Okay, sir.
my question is, what is the technology? What is the for farmers? If you say
Okay.
only one technology, a comprehensive technology to be
Okay,
followed to get a good crop with the sunflower nectar, to how to avoid single a technology package. What do you recommend as per your
sure, sir.
recent book?
I'll send in the chat box, sir.
Yeah, chat box you can
Okay.
write that what is the technology available with your university to
Okay,
manage
sir.
this one. Only one para, not
Okay, sir.
much.
Sure, sir.
Another
Thank
small
you, sir.
comment, Dr. Selvarajan from
Yeah, Suresh. Yeah, Dr. Suresh. Yeah, please go ahead. You ask. Joining from.
Sorry.
Yeah, Suresh. Yeah, we are giving you time. You can ask your question, please.
Yeah, it is a wonderful presentation. As I have been seeing this work, it is a very classical work. And in this time, a study like this is a classical pathology studies. I compliment the work has been done by Bharti Bhatt and team. My only comment is that in the classification of resistance or moderately tolerant or susceptible, I may not recommend to put immunity, because immunity, no breeding work is being carried out for immunity. Perhaps in the future, you may not consider to put immunity in the classification of resistance. So when you go to farmers, or when you go for extension people, or when you go to for any people, they only talk about resistant, tolerant, or susceptible. So I won't recommend to put immunity in the classification of resistance, number one. And second point is that you have done a wonderful work on the artificial inoculations to understand alternate host or correlative host. Do you see any natural infecting host in the crop field in your studies? And you can put in the chart box as rightly said in this point. So I don't want to take much time, but I congratulate the work being done by you, and it's a kind of a model study for other virus work.
Okay, sir. Thank you, Dr. Suresh Garu. I'll answer in the chat box.
Yeah. Please. Thank you, Dr. Suresh, and thank you, Dr. Bharti. And the next speaker is Miss Tirumala Raju Surya Sumas Chandana.
Yes, sir.
Madam, Dr. Bharti can close your this presentation.
Okay, sir.
Stop sharing, and I request Miss Tirumala Raju to share.
I started sharing, sir.
Yeah. You didn't keep it open again. Just click, double click.
Sir, is it visible now?
No, it's not visible. Is it visible in your computer?
Yes, sir.
Here, no. Sesa, Dr. Sesa, you help. Your entire desktop is visible, not the PowerPoint. Chandana?
Yes, sir.
you just double click that PPT which you want. Oh yeah, now it has come. Yes, go ahead.
Okay,
Only
sir,
7
thank
minutes,
you.
please. You restrict because there are many presentations are ahead.
Okay, sir. Good afternoon, everyone. Myself, TSS Chandana, postgraduate student from College of Horticulture in Department of Plant Pathology, Dr. YSR Horticultural University. My today's presentation on the topic is studies on pathogens associated with gummy ooze secretion in bottle gourd, Lagenaria caesareia. Gummy stem blight is a cucurbit rot disease caused by fungal plant pathogen Didymella bryonia. It reduces yields of edible cucurbits by causing stem vining and rotting of the fruits. Gummy ooze secretion was noticed on bottle gourd plants at College of Horticulture, Venkatramnagudam, and in the farmers' fields. The branches of vine showed splitting of bark followed by production of reddish gummy exudate. As per the current literature, 3 causal agents were reported for inciting gummy ooze secretion in bottle gourd. They are Didymella bryonia, Fusarium roseum, and Fusarium verticilloides. According to AICRP Vegetable Crops Report of 2018 to 19, it was recorded that the etiological agent inciting gummy ooze in bottle gourd varied from region to region. The agent was reported as Didymella and Fusarium species in Hyderabad region of Telangana, Aspergillus glaucus in Kalyanpur. A mixed infection was seen in IIHR Varanasi caused by Fusarium oxysporum, Fusarium verticilloides, and Rhizoctenia solani. Didymella and Fusarium species in Hesargata. Since the etiological agent was varying in locations, current experimentation was carried out for isolating the pathogen, proving the pathogenicity, and identifying by morphological and molecular methods in our locality. The symptoms which we have observed in our locality are splitting of stem and secretion of reddish brown gummy ooze, light brown colored necrotic lesions which later expand to U or V shaped. Oozing of the gum is seen on the stem part, later complete drying of bottle gourd vine, and at the final stage, entire death of the plant is observed. Objectives for the experimentation are collection of gummy ooze secreting disease samples, isolation and pathogenicity studies on bottle gourd, morphological and molecular characterization of pathogens associated with gummy ooze secretion, in vitro evaluation of native isolates of biocontrol agents, botanicals against the pathogen. Um, Methodology: scouting for infected samples exhibiting symptoms such as gummy ooze and bottle gourd in farmer fields as well as in experimental plots at College of Horticulture Venkatramnagudam was carried out. We have collected samples with brown to black discoloration on the stem and those which exhibit yellowish brown sticky fluid secreting out. Collection of disease samples was done. They were washed to remove adhered soil particles. Stem, bark, and leaf portions were separated using sterile scalpel. Samples were dried for 24 hours in order to remove excess moisture. Cut pieces were later sterilized with mercury chloride, 0.1% mercury chloride. Excess moisture was removed by placing the pieces in sterilized blotting paper. Surface sterilized pieces were then transferred to Petri plates, which are previously sterilized, and antibiotics like streptomycin was used to prevent bacterial contamination. These Petri plates were later kept at 28 degrees centigrade for 7 days in an incubator. As soon as the mycelial growth was visible, for identification and maintenance of pure culture was done by single spore isolation technique. After 7 days, we have observed that the isolated culture produced white, aerial, and fluffy mycelium. This is the pure culture plate of Fusarium species, and we have observed macro and microconidia of Fusarium species when stained with lactophenol dye. To test the pathogenicity of the isolated pathogen, we have done pathogenicity studies. For that, the cultured pathogen was mass multiplied on sorghum grains for soil inoculation. After 7 days of inoculation, pathogen multiplied on sorghum grains was added to soil in polybags containing healthy plants to test pathogenicity. Some are not inoculated with the pathogen and they served as control. After 20 days of inoculation, we have seen black and dark patches on stem, and as well as wilting and death of the bottle gourd is observed. The same culture was obtained when we have re-isolated the pathogen, thus proving the pathogenicity of the isolated pathogen by Koch's postulates. For identifying the targeted pathogen molecularly, we have done a molecular identification. For that, we have extracted fungal DNA and isolated the fungus by genomic DNA protocol. Later, amplification was done by using PCR. PCR was performed in Eppendorf master thermocycler with these set of conditions, the PCR was performed with the ITS-1 and ITS-4 universal primers. Each PCR reaction was set up with positive and negative control. After the reaction was completed, it was run on 0.1%. The PCR product was run on 0.1% agrogel and visualized by Ethereum bromide. Thus, we have seen a band between 600 to 700 base pairs region, indicating the amplification of ITS gene of Fusarium species. This is the sequence results obtained. This is the sequence we have obtained after the amplification. This sequence, when we have done the blast analysis, it has shown 100% homology with Fusarium equicity, confirming that the isolated pathogen was actually Fusarium equicity. This is the phylogenetic tree we have constructed based on the sequence obtained, and here we can see that it has exhibited 91% homology with Fusarium equicity strain. These were the first reports of Fusarium equicity as a pathogen in several cucurbit crops. Later, we have done in vitro evaluation of native evaluation of native isolates of some biocontrol agents, botanical signatures the targeted pathogen. We have isolated soil. We have isolated bacteria from soil. Serial dilution was done, and later colonies were plated for incubation. Picking up of colonies was done after 14 to 16 hours of incubation. Screening was done for antifungal efficacy. Out of 60 colonies screened, 4 bacterial strains were found to be highly antagonistic. When we have gone for identification of bacterial species, we opted for multi-top mass spectroscopy. The identification is done by direct spotting of bacterial colony and protein extraction from the bacterial colony. The results we have obtained as that two strains belong to Burkholderia contaminants, and the other two are Stenotrophomonas maltophilia and Pseudomonas aeruginosa. Amplification of bacterial 16S rRNA gene was also done for further confirmation. We have done the amplification through the following primers, and the gel image evidently showed the amplification of the 16S rRNA gene of the bacterial strains. Screening studies were also conducted with bacterial and fungal biocontrol agents. Among the bacterial and fungal biocontrol agents used, Pseudomonas fluorescens has shown highest percent inhibition among the agents used. Screening with the native isolates of bacterial strains was also done. Among them, Pseudomonas aeruginosa has shown highest percent inhibition. These were the broad spectral these were the strains which I have isolated from the soil, and along with the Fusarium equisetae, the targeted pathogen, I have isolated other pathogens such as Fusarium solani and Sclerotium ralphsi from the other infected bottle gourd plants, and has observed that these strains which I have isolated from the soil have broad spectral antifungal property, as they have shown strong inhibition to the mycelial growth. It clearly indicates that the strains has a strong antifungal property. Screening with botanical extracts was also done, when 7 botanical extracts were taken, and they were tested at different concentrations of 2.5%, 5%, and 10%. We have observed that Funica granatum has shown highest percent inhibition. Sclerotium ralphsi was also isolated, and it was screened with native bacterial isolates. We have seen that Burkholderia contaminants has a strong inhibition to Sclerotium ralphsi, such that the sclerotial bodies did not germinate at all in the presence of bacterial culture. Conclusions: we found that the pathogen inciting gummy ooze in bottle gourd in our locality is Fusarium equisetae. 4 bacterial strains were isolated from the soil which are having antifungal activity and were identified as Pseudomonas aeruginosa, 2 Burkholderia contaminant strains, and the other is Stenotrophomonas maltophilia. Among them, Pseudomonas aeruginosa is effective against the targeted pathogen. When tested with other fungal biocontrol agents, Trichoderma viridae has shown 66.69% inhibition to mycelial growth. When botanical extracts were used by poison food technique, Funica extract has shown highest inhibition to mycelial growth. Our future line of work is to the targeted pathogen will be further identified at species level by using other housekeeping genes such as alpha tubulin, alpha actin, beta tubulin, translation elongation factor 1 alpha. In vitro efficacy of bacterial and fungal bioagents was also done. along with botanical extracts. Further, we have to do pod studies and field studies to develop integrated decision module. Secondary metabolites, which are responsible for antifungal activity from the native isolates of bacterial strains, will be extracted and identified. Acknowledgement, my sincere thanks to my chairman, Dr. K. Seshakiran, Assistant Professor, College of Horticulture, Venkataramanagudam, Dr. Vaisar, Horticultural University, Andhra Pradesh, and Dr. Praveen Rahi, Scientist, NCMR, Pune, Syed Irfan, Research Scholar, NCMR, Pune. Thank you.
Okay, thank you,
Chandana,
Chandana, for your.
you didn't share. You didn't show that the sclerotium also. That also isolated. No, why you skipped that one? That slide you just.
Actually, it got skipped, sir. Sorry.
Okay, okay. You can
No,
explain
it
that
was
also.
visible. Yeah, she showed not the end.
No,
Yes,
first one, no, she forgot, sir. She just skipped that one.
sir, bottle guard,
Yeah.
along with the other symptoms, we have seen this gummy use where I have seen sclerotial bodies, red colored sclerotial bodies were also observed. It has also showing the same symptoms, gummy use symptoms.
Okay, thank you. Thank you, Chandana. The questions, I think I again request the people to post in the chat box and you may answer. My question is, okay, I have got one question. When you have identified many pathogens and you worked with only one Fusarium equisity, why not all other, is it a complex disease or it is a disease caused by a single pathogen? So this you give.
No, she
Then
didn't
you can
isolate,
give.
sir. No,
Okay.
it was reported elsewhere, but in our region we got only Fusarium equisity.
But not sclerosis also.
No, that too she got in our region.
Okay, okay. That's good. That's good. And really, it's a good work. And I think a future work will come out with a good integrated approach for managing this disease. Now, I request the third speaker,
She has
fourth
just
speaker.
carried out in just 2 months, sir, all this work.
Oh, yeah. It's very good, very comprehensive, and it's a lot of data you have obtained. Further, one has to work to get a real integrated management. You had to make, combining all of these things put together. We had to come out with the integrated management approach. Now, I request Susmita Rajkoda to present on cloning viral genome associated with leaf curl disease of Budd Jolokia. Susmita.
Yes. Good evening, sir.
Yeah. Share, please. Share your presentation. 7 minutes. You cut off this introduction and all. You can straight away go for your research part.
Sure, sir. Is it visible, sir?
Yeah.
Good evening, everyone. Myself, Sushmita Rajkua. I'm a postgraduate student in Assam Agricultural University, Department of Agricultural Biotechnology. So I would like to share about my master's research work here. And the title is Cloning of Viral Genome Associated with Leaf Curl Disease of Bhut Jolokia, which is Capsicum chinense jag. So giving a brief introduction of Bhut Jolokia, it has been thought to be a hybrid of Capsicum chinense and Capsicum frutescens, but recently it has been distinguished as a distinct species, Capsicum assamicum, and it is basically native and found in the states of Assam, Nagaland, and Manipur, and it is economically very much important because around 1400 to 1500 tons of fruits are exported annually just only from Assam. So the other names are O Morok and Naga Jolokia in the other states. The important properties of
Sorry, 1
Jolokia is
minute. Dr.
that
Sesa
it has...
please mute your this one.
Excellent.
Dr. Sesa, please mute. Yeah, go ahead, Susmita.
Yes, sir. So the properties of Bhut Jolokia is that the most importantly, it has been recorded as the world's hottest chili in the year 2007. But presently it is in the 7th state. But other than that, it is also widely important in the international market because of the capsaicin content it has, that is around 3 to 5%. So it is also been used in medicinal properties as painkillers or in cancer therapies. It is used as pickles in culinary. It has recently been used by DRDOs for biological warfare programs, and also traditionally it is used for repelling elephants. But recently there has been a lot of viral disease identification which has been reported in the Bijolokia cultivation in Assam, and it includes like leaf curl, fruit deformity, stunting and premature death, and low yield because of these viruses which has been reported. These are the tomato spotted wilt virus, the cucumber mosaic virus, the potato virus. So these are the RNA viruses reported. Yeah. Next, it has been found that there is vein clearing, vein thickening, yellowing of leaves in stunted growth, which are being reported by the presence of Gemini viruses, tomato leaf curl Bangladesh virus, and tomato leaf curl virus. Next, there has also been report of chili leaf curl virus. So these are the DNA viruses. So looking into the problem of these virus infectivity, we have come into determination of two objectives. One is the cloning of viruses associated with the Buddholokia leaf curl disease, and second is if there is presence of any satellites, then there will be the cloning of the associated satellites causing this disease. So we started with sample collection from different locations of Assam. We included 4 districts nearby the areas of Assam Agriculture University and the nearby districts also. So these are the photographs of some pictures which I have taken during the sample collection. So next is the flow which we have followed. First, we have extracted the genomic DNA using CTAB DNA extraction method, and we did the spectrophotometric analysis of DNA using nanodrop. The qualitative analysis was done through agarose gel electrophoresis, and the detection of the virus was done through PCR using both universal primers as well as gene virus specific primers. And then we went for cloning using the Topo TA cloning kit. After the cloning, we went from sequencing. Then we aligned the sequences. We did a blast search, and then we did the phylogenetic analysis. So these are the this is the universal primer which we used initially for the detection of the virus, and these are the conditions which we use for the reaction mixture. Next, these are the results which we got actually. We didn't get positive bands in all the samples we collected, but we got some bands. So we extracted these bands, we purified it, like we eluted it, and then we went for cloning it directly using the Topo TA cloning kit. So these are the conditions which we use, and it was a kit we used for the Topo cloning. And next is that we prepared the competent cells in the lab. We went for blue-white screening of the positively inserted fragments, and we again, for confirmation, we did a colony PCR, and here we got the bands positively which are desired. So next we went for plasmid isolation of these uh, colonies which we got. We collected the plasmid, and again for reconfirmation we did the restriction dissection, and here we can like you can see, after the restriction dissection we got the 3.9 kb fragment and the 800 base pair fragment, which is our which was our inserted sequence. Next, when we went for sequencing of these isolates, we did not get what we expected. Instead, we got 78% identity with that of the capsicum enum. That is, we got similarity with that of the chili plant instead of getting the similarity with the viruses. So what the next strategy we went is that we designed specific primers only for the chili leaf curl virus using the Prime 3 software. And we used these reference sequence, which was already present in the NCBI. So we made tests of primer specific primers, virus specific primers, and we used these conditions for doing the PCR. This time also we got multiple bands, and from these bands we extracted desired like around 500 base pair fragments from here, around 500 base pair fragments and around 1000 base pair fragments, because we don't know that which one would work and which one is our desired virus sequence. And from the second primer pair, we got these 3 bands which were distinct, and we eluted them in the gel. Then we directly went, instead of cloning this time, we directly went for sequencing for confirmation. And from the 4 samples, we actually got partial gene fragments, viral sequences of around 460 to 418 base pairs. And it showed 98 to 96% identity with that of the chili leaf curl virus isolate this time. So we went for finding the ORFs online, and we got from one biggest fragment which we had. From there we got the replication enhancer protein with 96% identity and 100% query coverage, so that we were fully 100% confirmed that yes, this was our chili leaf curl virus isolate which we got. And doing a phylogenetic analysis with other available leaf curl viruses that has been reported earlier in Chile, we got our isolate in the cluster of the old world, that is which has been reported from the old world, and the other could be differentiated into the new world. Next, it was also reported that other than chili leaf curl virus, there was also report of tomato leaf curl virus. So we went for detection of these also using tomato leaf curl virus specific primers, but we did not get any specific detection of any tomato leaf curl virus, although we got some smears and some bands, but their sequencing also did not result in any virus similarity. Instead, we got any capsicum enum plant related sequence itself. Coming to the objective 2, the importance of having a satellite is that it is mainly helping the virus to replicate inside and produce a higher amount of symptoms inside the plant. So we went for finding if there is any presence of satellites. So we went for 2 satellite specific primers. One is DNA A 101 102 and beta 101 102, and using these conditions, but we did not get any satellite presence. So our second objective was negative. So the summary of the work whichever I have done is that the universal primers were initially used for detection. Then the amplified PCR products were eluted and cloned to topo TA cloning vector. The cloning was confirmed using colony PCR and through restriction digestion. And the recombinant clones were sequenced and analyzed, but the viral sequences were not found. Then we went for virus specific primers for doing the detection. We designed 2 pairs of specific primers. We amplified the products, and then the sequences were blast analyzed, followed by an ORF finder for phylogenetic analysis.
Okay.
Then tomato leaf curl virus primers were also used, but the virus was not detected. Similarly, the satellite genome was also not detected. So the conclusion was that after the research, because we only had 2 years of time to complete, we could not go for the whole genome sequencing. Instead, we ended up with having only 4 partial sequences for chili leaf curl virus. And from the primer pair 1, we obtained 2 positive from the samples, and from primer pair 2, we also got 1 positive. Similarly, it showed 90% similarity with the chili leaf curl virus isolate, and it was the genome was showing similarity with that of Oman. So that may be because we only had the partial sequence. We did not get time to cover the whole sequence. Again, the satellite genome was not detected. So that was all about whatever I could connect. And I would like to acknowledge my major advisor, Dr. Bashant Kumar Bora sir, my advisory committee members, M.K. Modi sir, Dr. Priyabrata Sen sir, Dr. Palash Devnath sir, Dr. Sunaina Rathi ma'am, and Dr. Hanu R. Pappu. The part of this work, I got the opportunity to complete it at the Washington State University by availing the Khorana Program of Scholars 2016. And I would like to thank Dr. Hanu Pappu sir for providing me the lab and giving me all the opportunity to learn all the techniques. I would like to thank all my department members and my faculty members, and thank you so much.
Okay, thank you, Susmita. I think Dr. Bilal has asked one question in chat box. You can answer. Can you close this? Can you close this screen? Yeah, please stop sharing.
Yes, sure, sir.
Yeah,
yes, sir.
It's a good work, and your presentation was very good, and you could able to relate the leaf curl virus with your Buergelacia leaf curl disease. And you would have gone rolling circle amplification straight away. You would have got the entire genome. So rolling circle amplification followed by a cut with some enzyme, and you would have cloned it, and you would have got the entire genome. Anyhow, in future you can do that work. Kindly answer the question posted by Dr. Bilal. And now the next speaker is Dr. Srisa Yeturi. Srisa Yeturi, are you ready?
Yes, sir. I'm ready,
Yeah,
sir.
yeah. Please share and take only 7 minutes. Sir. Don't take more time.
No, sir. I'll be sharp, sir.
Yeah, please.
Thank you, sir. Yeah. Good evening, one and all present over here. And I'm here to present my thesis topic at part of my research, rapid and specific detection of Pernosclerospora sorghii in maize seeds by conventional and real-time PCR. I'm Sirisha Yeturi, Department of Plant Pathology here, working at Dr. YSR HU. And the thing... So is my... is this
go for slideshow? Go for slideshow.
Yeah, yes, sir. Yes. Sure. Yeah, as we all are well versed with about maize and its importance, that is as it is of queen of cereals, and it is a third after rice and wheat in its production, and it is very important for its quality protein maize with the world's largest maize area of product like 9.09 million hectares and all. And the yield of maize has been reduced because of many diseases, and among them the maize has been reported with as a host for 8 Pernosclerospora species. Among these, which are caused by Heteropogony, Maydis, Misanthe, Philippinensis, Saccharine, Sorghii, Spontanea, and Ericleae. Among these, I have been concentrated on Pernosclerospora sorghii, which is the main causal organism for both maize downy mildew and also sorghum downy mildew. Here, these are the maize downy mildew symptom, and it is an obligate parasite which shows downy growth on the underside of the leaf, and also at the later stages we can also see the crazy top symptoms. And these symptoms will be very vast and severe, disastrous with its systemic infection. And the conditions which are favorable, we are already well versed, that is it is very peculiar with the cool and humid temperatures, humid conditions. And this is the like microscopic picture of Pernosclerospora sorghii. And these I mean these are for conidial inoculum production. We can also have even on petri plates and also in large scale in beakers and for sporulation. For sir, is this any disturbance, sir? From my side, is there any disturbance? Okay, and this is the stage of inoculation where we have to go for inoculation at 2-leaf stage, and this is the symptom which is developed at 10 days after inoculation. And this maize downy mildew symptom, we have to maintain the culture even mainly under glasshouse conditions. And this is the first chlorotic symptom which has been developed after inoculation. And actually we have collected like 20 isolates of maize and sorghum where we have developed a scar marker for the detection of Pernosclerospora sorghii in maize seeds and soil as it is of seed burn, and also it has its infection from soil-borne ooze spores. And this is the primer which has been like a total of 30 RAPD primers we have taken. Among them, like OPE-D16 has shown a differentiating band for like a detection of base orgae, Penosclerosporus orgae, and this is the sequence which has been generated, which has been amplified, and it has been cloned and cloned into PGMT vector for the plasmid isolation. And this is the total sequence of Penosclerosporus orgae, and this primers, forward and reverse primer, has been submitted even at GenBank with the GenBank with the accession number KR02846. And these are the two primers, forward primer and reverse primer. These are SCAR primers which has been developed from this RAPD region, and these SCAR primers has been validated by again collection of extra isolates from maize and sorghum for the standardization of PCR assay. And these all maize and sorghum isolates has been collected in and around Coimbatore and also other areas of Coimbatore. And this is the clear band which has been clear amplification of PCR amplification of Penosclerosporus orgae using SCAR primers. And a peculiar band of 305 base pair has been amplified, and this SCAR primers has been validated, and specificity also has been checked with the check control. And this is the sensitivity, even the detection can go near to 1 nanogram. And this is, and the first one is for fungal DNA specific sensitivity, and this is from infected seed sensitivity. It has even gone near to 5 nanogram. And these seeds, this is the detection of Penosclerosporus orgae from downy mildew infected plants directly collected from infected plants, seeds collected directly from infected plants, and the detection has been validated. And these are the total of maize seed samples which has been collected from different different seed samples from different areas, like even from IIMR, Hyderabad, Coimbatore, Tirupur, Udumalpet and all. These total maize seed samples were collected DNA, isolated DNA, and we have gone even for PCR, even by conventional PCR and also through real-time PCR. And this is the conventional PCR gel picture, whichever we are seeing here. And this is a total of like 70 samples we have collected and we have validated. And this is the real-time PCR assay which has been which has been amplified, and a total of standard curves are also maintained, and detection of Penosclerosporus orgae has been validated using SCAR primers even from infected seed samples. And this is the correlation standard curve which has been correlated with the check and also from the maize seeds. And a correlation coefficient of 9.96 was plotted, and this is a melting curve which has been ranged temperature at 95 degrees, which has been correlated even from seeds and also from fungal DNA and also from this soil, sick soil, whichever the ooze spores has been detected. And this is the validation of this gel picture, which has been actually this is from seeds and also from the soil. Sick soil has also been detected with the SCAR primers. This is what we have done as a part of research. And at the last, the SCAR primers which has been sequenced has been validated even at the quarantine stations. Okay, and that is all my work. Thank you for this opportunity which has been given, and I sincerely acknowledge the Department of Science and Technology for my INSPIRE Fellowship, and also Department of Plant Pathology, TNA Coimbatore, and also my Department of Plant Pathology, College of Agriculture, Horticulture, Anantarajpeta, Dr. Vaisar Horticulture University. Thank you one and all for listening to me. Thank you. Thank you so much.
Thank you, Dr. Srisa. Very good work you have done, and doing a detection for this Penosclerospora is very Penosclerospora orgae is very important. And again, you detected in seeds. That's very good one, and it's a good work.
Thank you,
and now I just invite the next person. Arvind Karni is there. Arvind Karni. Arvind. Okay, if Arvind is not there, the next person is Dr. Balram. Dr. Balram. Dr. Balram. Okay, Dr. Balram is not there. Dr. Tota Nagalakshmi. Dr. Nagalakshmi is there.
I'm here,
Dr.
sir.
Nagalakshmi, please, you go ahead with your presentation on genetic diversity of alternaria podi isolates causing purple leaf blotch of onion in Andhra Pradesh, Karnataka and Telangana. Okay. Please share your presentation. You can go ahead.
Good evening, everyone, sir. I am Nagalakshmi, scientist, plant pathology, working at Anantarajpeta, under Dr. Vaisar Horticulture University, sir. Today's my topic is genetic diversity of Alternaria pori causing the purple leaf blotch of onion across the different states like Andhra Pradesh, Karnataka, Telangana, where it is a very serious problem in onion growing states, sir. Then sir, these are the major onion growing states in India. Majorly, it is grown in the Maharashtra, followed by the Madhya Pradesh, Karnataka, Gujarat, and so on. So, coming to the area, it is every year it is increasing, sir, but yield is decreasing because of this disease, which is causing a lot of, it is a devastating disease causing 50 to 100%. For example, now weather is, for the last 1 month, weather is very congenial for the development of the disease, which is causing a total loss to the onion growers across the Andhra Pradesh and Maharashtra and other Karnataka also. So that much severe this disease is, sir. That's why I have taken this as our research work. And these are the different areas, mandals growing, onion growing mandals in YSR Kadapa district, sir. And these are the Kurnool is a very potential area for onion growing. And this is the map showing the different mandals, important mandals growing the onions. And this is a peculiar correct symptom of this purple blotch. First, it appears like white spot. Then it develops into a dark brown color because of its sporulation. It is a dark brown, so it produces dark brown spores. This is the typical symptoms showing on a single plant. Here you can see the severity of the disease in background. First, it appears like a point like a pinhead size white spots. Later on, elliptical, these zonate spots will be appear on the severe conditions. And this black sooty mold-like substance is nothing but a sporulation, which produces millions and millions of spores and devastate the entire crop if you don't react within the time, sir. That much severe it is. So these are the field symptoms. After incubation, we have isolated. We have observed the mycelial growth on the each and every spore symptoms, sir, even a spot. So from there, we have isolated the pathogen for the purification of the pure to get the pure culture. These are the different spores observed under 100 microns. Microscopes, and these are the germinating spores. And this is the pure, third one is the pure culture. We got the different growing stages of these conidial stages, and serial these produces in a series of series these spores conidia. And coming to the pathogenicity, we have conducted survey across the three states, onion growing states. Among those, we have selected 15 isolates on particular regions, like up to 9 isolates from Andhra Pradesh, 4 from Karnataka, 2 from Telangana. These isolates collected, purified, and pathogenicity study and virulence studies also we have taken up. And by seeing these virulence studies, these are some isolates are highly virulent, and some are moderate, and some are just virulent. By using this chart, we have categorized these 15 isolates into 3 categories. These Maidukur Kadapa isolate is highly virulent, and Anantarajpeta also, like another one Nandikotkur. These are the highly virulent isolates we have found through this virulence test. Then we have cultured, and different cultural studies, morphological studies were also conducted. And this is a table showing the different conidial length, width, and beak length also varying from the isolate to isolate. And the number of septa also differing, like horizontal septa ranges from 3 to 13, whereas vertical septa 0 to 3. In some cases, there is no vertical septa. In some cases, up to 3, 5, 6, like that it has gone. In some cases, oblique septa also we found. later on we have and on PDB potato dextrose broth. These are cultures for groom and for the isolation of the DNA, and this mycelial mat was separated and DNA was isolated by using CTAR method. And for molecular confirmation, we have used IPS 1 and 4 these primers, and following then we have confirmed by blasting the sequences. These are the different programs we have used for the both ITS and SSR primers. ITS 1 and 4 were used for only identification for confirmation, but this by using this ITS 1 and 4 we cannot study this variability across the different regions and different isolates. For that purpose we have gone for the SSR primers. From the published information, we took the 10 SSR primers and programmed by using different annealing temperatures. And this is the gel picture showing the all are confirmed by using this ITS 1 and 4 and grouped also. Then later on, this is we have gone for the SSR primers screening to know the genetic variability across the different groups of isolates of the Alternaria pori. And these are the forward and reverse primers we have used. And these are the gel pictures. In some primers, SSR primers didn't get any amplicons, and in some cases found specific bands like this, like SSR 7, 8, 9, 10. All these cases we found this some specificity for some isolates. And then we have calculated polymorphic information content using this formula, where this polymorphic content is very useful for identification of and study the genetic variability across the pathogens like isolates. The population this range PAC value ranges between 0 to 1. 0 means there is no variation, whereas 1 is there is a highest variation, the maximum variation between the isolates. That is the use of these peak values. So by using this formula, we have calculated the peak values for all the 10 isolates. These particular table shows the entire work done for the 15 isolates using 10 SSR primers. So coming to this, some of the primers showed polymorphic bonds and some unique bands were found. Totally, we could get 105 bands. And peak values, coming to peak values, there is a genetic variation showed by seeing these peak values, we can understand that there is a variation. Then proximity matrix was also calculated and then it shows that there is a similarity coefficient range between 0.4 to 9 across the 15 isolates. The results of a pairwise combination indicated that the highest similarity coefficient 0.916% between the isolates like AP4 and AP9. These are isolates obtained from the different onion growing regions of Andhra Pradesh. Whereas some isolates, there is a lot of difference, different genetic variability observed between Karnataka 4 isolate and TS2 exhibited the lowest degree of similarity, means there is a lot of variation between these isolates. So by using this SSR primers, we have studied this genetic variability. So these are the conclusions. First, we have collected 15 isolates and purified DNA isolated. Morphological studies were done, and among by using the ITS 1 and 4 primers, the confirmed molecular level and sequence similarity studies were also conducted, and using neighbor joining method, proximity studies conducted for and isolates were grouped into 2 clades. Then genetic variability studies also conducted by using the 10 SSR primers, which showed that SSR out of by using these 10 primers, we could get 1 or 2 loci. Maximum number, average number varied from 0 to 27 with an average number of bands per primer was 8.7. The 2 SSR primers, that is SSR 7 and SSR 10, gave unique bands for some isolates like AP, Andhra Pradesh 6 and Karnataka 4th isolate. Likewise, SSR 4, SSR 10 gave unique bands for AP 2, 3, 6 and 9 isolates. Then these proximity studies were conducted and the similarity coefficient was showed that there is a closer similarity observed in between these AP isolates and there is a lot of genetic variability between Karnataka 4 and PS2. These are the actual results we obtained from our genetic variation studies. These results can be utilized for developing management strategies for develop new resistant cultivars through marker resistant selection. Sir. These are the future plans of our research. Sir. Thank you very much for hearing.
Thank you.
Anand for giving this opportunity. Sir. Thank you.
Thank you, Dr. Thank you, Dr. Nagalakshmi, for giving a wonderful presentation on alternaria puri, the purple leaf blotch of onion. Suresh wants to have one question. Yeah, quick question, Dr. Suresh, please.
Yeah, one question. I think it is a very important studies in onion. As a purple leaf blotch, it is a very important disease in entire India as well as in the world. So the implication of this studies is the most important. As you say that virulence studies, that you have not mentioned perhaps which host, which variety you have used, number one, which environmental condition you have used, because when you have different environmental conditions and different variety, the variability of symptoms, the virulence would vary. That is important to decide. And number one. Number two, do you have any protocol which you can think of in-house screening germplasms to recommend for the breeding program? We cannot just directly go to marker studies, marker development, because there is no resistance for this particular disease in the world. As I work in some of the key onion breeding program. So keeping in that mind, perhaps we need to first standardize the protocol and also look for land race, look for key resistance sources. That would be first beginning for a breeding program. Then the later management studies, management practices will come. Perhaps you can keep these points in your recommendation for the future studies.
Okay. Thank you, Dr. Suresh, for giving two important comments on this paper. And if you don't mind, you can write that comment on this chat box if you could able to.
Yeah.
So we'll add into the proceedings of the this session. So thank you, Nagalakshmi. Let us go for next talk by Dr. Sabha Bande. Dr. Sabha Bande. Dr. Nagalakshmi, you can close this. Nagalakshmi, you can close this screen. Yeah. Oh.
Excuse me, sir. Sir, I'm Dr. Madhavi. Shall I proceed after this presentation, sir?
No, 1 minute. Dr. Sabha Bande is there or not?
Yeah, yes, sir. I'm present here.
Yeah, Sabha Bande, please go ahead. Madhavi, wait, please. Just
okay.
wait.
Okay, thank you.
Okay, sir. I'm not able to share my presentation.
Why? There is a green arrow mark in the down,
Yeah. I'm clicking over there only, but it is not getting shared.
Okay, then you do.
Yeah.
You mail to Selvarajan sir or me?
Or Sesa, you can mail to Sesa Gran or me.
Okay,
You mail. Let us look for the next. Chatana is there?
Yeah, it got shared, sir.
No, not it. No.
Yeah. Now it's okay.
Coming. Yeah.
Showing, sir?
It's coming.
No, it's loading. It's fine. Okay.
Yeah. You go for slide mode. Full slide mode you can go.
Yeah. Showing?
Yeah, it's visible now. You can go ahead with the presentation. Kindly complete within 7 minutes, huh?
Madam, slideshow. Go for slideshow mode.
Okay.
Down, slideshow is there, no? You go there.
Okay, sir.
Yeah.
Good evening, one and all. Good evening, one and all. I am Dr. Sabha Bande, Assistant Professor, Plant Pathology, Cost, Shalimar. So I will be, my topic is here molecular variability of Sphaceloma ampelligens of India. So the objective of this study was to study the morphological and cultural variability of Sphaceloma ampellinum and to study the molecular variability of Sphaceloma ampellinum. So giving the brief introduction of the importance of grapes in India, as you know, that grape is a very commercially important fruit crop of India, and grapes are cultivated in an area of 117.6 thousand hectares with a total production of 1,234.9 thousand tons in the country. So it is making remunerative commercial farming enterprise. Giving the current status that is by the Public Information Bureau 2012, India export totally of 52,000 dry grapes that value 40.1 million USD dollars. So in Kashmir as well, this grape is an important crop as it is grown over an area of 427 hectares with a production of 742 million tons. And owing to the agroclimatic conditions of the crop in the region of J&K, it is harvested in the month of August when it is not available in the rest of the country. So many diseases are they are impairing the production potential of the grapes all over the Kashmir, and some of the major diseases are fungal diseases are like downy mildew, powdery mildew, and anthracnose is one of the important diseases that hampers the full production potential of this of the grapes. So give just the background of this research. I have to get because hello.
Yeah, please.
Are you able to hear me?
Yeah, your voice went off, but now you can go ahead.
Okay, sir. So I was talking about the symptoms of this grape anthracnose. Just, sir, on the twig, it comes as a canker, and on the berry, it comes as a bird-sized spot.
Yeah, next. Yes.
So, grape anthracnose. Hello, sir. Sir, actually,
Yeah.
here 2G
Please, go ahead.
internet services over here. So I think.
Sabha, you can go ahead.
So on the okay, sir. So this is the 40 days old brownish white colony, and these are the spores, greenish conidia. And we have done the molecular characterization of the isolated pathogen by the forward primer and the reverse sequence of the primer. And expected amplicon was at the 500 base pair that you can see in this picture. So various isolates were isolated from the different regions of the, and since Illustrinae amplina very difficult to isolate a pathogen, so we isolated 20 isolates from different parts of the Srinagar, and the morphological as well as the molecular isolates. And you can see that mostly the isolates were wrinkled and some were raised, and some of the isolates no acerola was present, and the spores were of very small size. These are the pictures of the different isolates. This was the pathogenicity test that was done on the detached leaf method, as well as on the detached twig method. So here we got the number of polymorphic bands, and this is the percent polymorphism in different primers that was seen. ISS are profiling of the 20 isolates of this Phaseoloma. And for the different primers, we got the peak values and the conclusion was that a significant morphocultural variability was found in Phaseoloma amplinum, but phenotypic characters are often difficult to observe and analyze. Peak values for ISSR marks used ranged from 0.31 to 0.37, which depict the efficacy for application in genetic studies. High genetic diversity in Phaseoloma populations may be attributed to several evolutionary selection pressure that might be active on the pathogen populations. Some of the isolates showed similar genetic and morphocultural grouping, and as fungus is highly diverse, effective management strategies like resistant varieties and fungicides are required for limiting new virulent pathogen genotypes in the pathogen population. Thanks.
Oh, thank you. Thank you.
Sorry for the inconvenience, sir.
No problem. Thank you, Dr. Sabha. You have presented very nice work, and I know this pathogen, handling this pathogen is very tough, and we have isolated quite a good number of 20 isolates, and it's very good work. Of course, your internet was giving some trouble, but we could able to catch up the what you want to say to us. Okay. Thank you. The next one is Dr. Chaitana. Is there?
Yes, sir.
Dr. Chaitana, please, you can go ahead with your presentation. Morphological and cultural characterization of Cytalidium thermophyllum isolated from button mushroom compost. Yeah, please. Dr. Sabha, you can close this screen. Dr. Sabha, please close the screen.
Yes, sir.
Yeah, kindly close the screen and allow the Chaitana to.
Yes, sir, yes.
Yeah.
Sir, is it visible?
I've closed.
No, she has to close. Yeah, now you start. Yes, good, visible. Go ahead.
Sir, wait. Very good evening to one and all. My name is Chetna Jangai, student from IGKV College of Agriculture, Raipur, Chhattisgarh. Title for the oral presentation is Morphological and Cultural Characterization of Scytidium thermophilum.
मैम आप लोगों ने कितना सुना और कितना छोटा?
Sabha, can you mute your this one mic? Yeah.
So title for this oral presentation is Morphological and Cultural Characterization of Scytidium thermophilum isolated from button mushroom compost. Here is an introduction. Thermophilic fungi are the small assemblage of eukaryota which have a unique mechanism of growing in elevated temperature above 40 and below 60 degrees centigrade. The thermophilic fungi are those microorganisms that can degrade by secreting several enzymes like cellulase, hemicellulases, and hydrocellulases. In mostly Chhattisgarh, mostly long method of composting is being used for the preparation of compost for the cultivation of button mushroom. Compost is generally is a product of fermentation bought by many organisms, including actinomycetes, thermophilic, and thermotolerant fungi. Majorly, there is two objective for this. First is isolation of thermophilic and thermotolerant fungi from the button mushroom compost. Second one is identification and cultural characterization of thermophilic microflora isolated from button mushroom compost. For this purpose, isolation was done under the natural condition. For this, the isolated plates were kept naturally in the heaps of compost. This is the compost prepared for the button mushroom cultivation. Here, the inoculated plates were kept in the box and were kept 1 to 2 inches deep from the upper surface of the compost. Here is a graph given in which different temperatures were given from the very first day to the 28th day of the time duration of the compost preparation. For the purpose of isolation, two methods were being used. First was the Waxman direct inoculation method. For this, four different medias were used: YPSS, YGA, potato dextrose agar, CZA medium, in which Zapekdox agar proved has a maximum number of microflora, where the least was observed from the potato dextrose agar media. And the second method was used was direct dilution plate technique. In this technique, 3 concentration were used: 10 to the power minus 3, 10 to the power minus 4, and the 5 concentration, in which 10 to the power minus 3 proved maximum number of thermophilic and thermotolerant fungi, and least were observed in the 10 to the power minus 5 concentration. Here Scytalidium thermophyllum has a maximum number of colony in between thermophilic and thermotolerant fungi. These are the several thermophilic and thermotolerant fungi which has been isolated from button mushroom compost, which has Absidia corymbifera, Scytalidium thermophyllum, Chitomium senegalensis, Thermomyces lanuginosus. In this Chitomium senegalensis, this is the perithecia, dark colored, and this is the spores of the Chitomium, and this is the species of Thermomyces lanuginosus. These are more several thermophilic and thermotolerant fungi which has been isolated: Humicola fuscoatra, Mycelioptera fergusi, and Humicola insolens. Others were the Aspergillus species such as Sidoi fumigatus, Aspergillus niger, and Aspergillus terreus. Here is the pictures of Scytalidium thermophyllum. First picture is after the 12 days of incubation. This is very matured conidia, which is from light brown to dark in color. And here is a after 6 days. This is the picture microscopic pictures of Scytalidium thermophyllum. This is the stain color. In generally, it is a colorless. Next is the cultural characteristics of Scytalidium thermophyllum. For these 4 medias, different medias were used, and in these 4 different medias, Scytalidium thermophyllum is being grown. In this, malt extract agar proved best for this for the growing of Scytalidium thermophyllum, and the least were observed in the Zapekdox agar. Next was done for the effect of temperature on Scytalidium thermophyllum, in which maximum growth and good growth was observed on the 40 degrees centigrade. In this, sporulation was also observed, and others in the 25, 30, and 35 degrees, and slow growth is being observed and no sporulation is being seen. Next is for the effect of different grains on growth of Scytalidium thermophyllum. For this, sorghum, grains, kodo, wheat, and paddy paddy is being used. For this, best was the wheat grains, in which it seemed to be fast growth, where least growth was observed in the kodo grains. For summary for this is a dilution of 10 to the power minus 3 concentration was proved best for the isolation of maximum number of thermophilic and thermotolerant fungi. And the Zapendox agar media was the better medium for the isolation of maximum number of microflora by Voxman direct inoculation method. And for the growing of Scatteridium thermophilum, malt extract agar medium was found superior above other. Fast growth and osmorylation of Scatteridium thermophilum was observed at 40 degrees centigrade. And the future prospect of this is development of suitable cost-effective method for mass multiplication of Scatteridium thermophilum. The main purpose for this is to reduce the long-term method for the composting for the growing of button mushroom. So this can be done further, and its molecular analysis enzymatic activity can be also done. Thank you.
Thank you for a very fast, quick presentation. It's very good presentation and nice PPT slides were there. Thank you, Zetana. Since there are 2 people absent, so we are giving a chance to another 2. Madhavi, are you ready?
Yeah. Yes, sir.
Madhavi?
Yes, sir. I'm here, sir. I'm here.
Yeah, please go ahead with your presentation.
Yes, sir.
Zetana, you please
Yes, sir.
stop sharing your screen. Can you stop sharing your screen? Yeah. Madhavi, please share your screen.
Yes, sir.
We have another 4 poster presentations are there. I hope people are there. Poster presentation.
Yes, sir. Shall I start, sir?
Yeah, please go ahead. Only 7 minutes.
Yes, sir. Yes, respected chairman of the session, dear colleagues and dear participants, a very good afternoon. I'm Dr. Madhavi, working as scientist, seed pathology, seed research and technology center, Professor Jayashankar, Telangana State Agriculture University, Hyderabad. I'm here to present my presentation on impact of seed discoloration on seed health and parameters in soybean. As we all know, like soybean is known for its high protein content for nearly about 38%. And if you see globally, the major countries which produce soybean was United States, and Brazil, Argentina, China, and India. In India, the crop is being cultivated in states like Maharashtra, Madhya Pradesh, and parts of northern Telangana. And under Indian conditions, for the past 2 to 3 years, germination has become a major constraint in soybean production. And excuse me, sir. There was some disturbance, sir. Yes, under Indian conditions, the germination soybean has become a major constraint for its production, wherein seedbed infections is one of the key factor playing in the loss of seed viability. For this reason, and here one more thing I want to add, like seed-borne pathogens. In its expressions, in its symptom expression, usually they cause discoloration on the seed. So for this purpose, the tolerance limits of seed discoloration on germination or germinability in soybean has been taken up by artificially inoculating the discolored seed in a healthy seed lot. For this purpose,
the screen is not coming, Madhavi.
Sir, I have been shared, sir.
No, it's not opening. No, it's a double click to enter full screen mode. Nothing. It is. Are others able to see? I'm not able to see.
Is it, sir?
No. Can you stop and again redo it?
Sure. Irana? 1 second. Sir, now it is visible, sir?
No, you stop sharing and again you redo it. That
Okay.
will be.
Yes. Please share. Okay. Sir, is it again? It is fast.
No.
It is not coming. Sir, now it is visible, sir?
Yeah, it is visible. You can continue.
Visible. You can go ahead, please.
Yes, sir. For the past 2 to 3 years, germination has become a major constraint in soybean seed production, wherein seedborne infections is one of the major factor playing a key role in loss in viability of seed. For this purpose, yes, actually, seedborne pathogens as a part of their symptom expression, so they cause discoloration on the seeds. So for this purpose, the tolerance limit of seed discoloration for germinability in soybean has been taken up. Here, artificially discolored seed was mixed with the healthy seed lots, and the studies were taken up by studying 400 seed per replication in each category. Here, the technique adopted was paper towel method, and the seed quality parameters such as germination, vigor, seed infection, and seed rot were studied. And here, based on visual observation, discoloration of the soybean seeds were categorized from 1 to 6 categories. And here the healthy seed was graded as 1, and the rest of the categories based on the discoloration. And here mainly to know the correlation between the germination and seed infection. Here category 1 means out of 400 seeds, as I said, but replication 400 seeds will be evaluated. Out of 400 seed, no disease or discolored seed was mixed in category 1, whereas in case of category 2, less than 1% means out of 400 seed, where 3 discolored seeds have been mixed. Likewise, category 3, 1 to 25% discolored seed were mixed. Category 4, 26 to 50%, and category 5 with 51 to 75% discolored seed, and finally under category 6, 76 to 100% discolored seed. So here, actually, for execution of this particular experiment, the soybean seed sample has been procured from the Kharif harvester produced during the year 1819 and 1920, and the 2-year study have been revealed significant differences between the seed discoloration categories for seed health and quality parameters of soybean, such as germination, seed infection, seed rot, and vigor index. And here the seed germination, if you look, this pooled data, the seed germination in both the years, the seed germination as well as the vigor gradually decreased with the increase in the seed discoloration. And while there was a corresponding increase in the seed infection as well as the seed rot. Here, in case of soybean, according to Indian mean seed certification standards, germination should be above 70 or above. So here, the categories, the germination recorded by the category 1 with 0 discolored seed and with 91.13%, and category 2 with less than 1% seed discoloration has recorded 87.25%, meaning with at above Indian mean seed certification standards. And here, if you look at these categories, the vigor also was 2370 and 2206. However, at these 2 categories, means 1 and 2 category, if you see the seed infection and seed rot levels, relatively or significantly lowest. However, if you move on to the categories from 3rd to 6th, yes, here there was a significant or gradual decrease in the germination as well as a vigor index, and gradual increase. I mean, there was increase in the seed infection and seed rot. Here, except for the categories 1 and 2, the rest of the category samples have recorded below seed certification standards of germination. Then here, overall means across the seed discoloration categories, here we could observe the seed infection and germination was negatively correlated. And these are a few photographs performed. Like yes, this is the less than 1% seed discoloration had good germination, and as a discoloration was increased, there was infections as well as rots have been increased. And these are the photographs wherein the seed were infected with the different fungi. Here the Aspergillus flavus like, and this is the Fusarium, Macrophomina. Again, these are Colotrichum, Cerulea of Colotrichum. Again, the Fusarium, Macrophomina and all. And these are some of the photomicrographic pictures of the fungi what we have been isolated, like different fruiting bodies and conidia of the Colotrichum, and Macrophomina, Phomopsis pycnidal bodies, Fusarium sporodochium, and conidia, micro and macroconidia. Then Alternaria and Aspergillus different, and even we could see the purple stain on, which have been infected by the Cercospora kikuchi. And finally, I would like to conclude, like in any crop, if you take, germination plays a key role in maintaining the plant stand for increasing yields, especially for crops like soybean, where germination has been becoming constrained because of, where seedborne infections are playing a key role. Here, from our studies, it has been concluded like soybean seed categories 1 and 2, which has recorded germination of above IMSCS levels with less infections and seed rots can be opted for sowing purpose for getting good stand and increased plant stand. In addition, I would like to add, like for any crop before sowing, seed, practicing compulsion practice of seed treatment is very much advised in order to prevent the seed and seedling rot diseases with any of the available seed dressing fungicide. So thank you for giving me this opportunity, sir. I thank you, the convener and chairperson of the session.
Thank you, Madhavi, for quick presentation. And can I ask the next speaker to talk?
Yes, sir.
Yeah.
Dr. B. Pushpavati, sir.
Yeah, please go ahead.
Yes,
I don't have your list. Yeah.
Sure, sir.
Pushpavati, yeah.
Yes, sir.
Hello. Good evening to all of you. Hello.
Yeah. You are audible. Please go ahead.
Okay.
Please share it, your presentation. Oh, somewhere it's going. No, this is not your presentation. Just close this. Yeah, close this window and go for your PPT.
Yes, we're trying, sir.
Madhavi, you help?
Yes, I'm helping, sir. Yes, sir.
Now got it,
sir.
sir. Sir, are you able to see now?
Yeah, please go ahead.
Respected chairman, sir, of the session, my dear colleagues and dear participants, now I am here to present a piece of work done in our laboratory during 2016, 17 and 17, 18. And I am going to present my research work on detection, location and transmission of seed-borne Alternaria sesame in sesamum. Alternaria sesame is known to cause leaf spot and pod blight disease in sesame. And we all know that it is a serious problem in sesamum wherever sesamum crop is grown. Especially in our state, the disease is very seriously occurring, and it is causing losses even up to sometimes 40% losses in seed production plots. That is the seriousness of the disease. And because of premature defoliation and that which leading to loss of capsules, this much yield reduction has been experienced in sesamum crop. And keeping this in view, its seed-borne nature of Alternaria sesame has been work has been conducted on proving seed-borne nature of Alternaria sesame in sesamum. For this, an experiment with an objective of determine the seed-borne nature and transmission of Alternaria sesame has been conducted in our laboratory. For this purpose, a total of 32 seed samples were collected from farmhouse fields as well as from the seed production plots of Agriculture Research Station Jagichal, that is one of the research stations of PJTSAU, and from all these samples, 400 seeds were evaluated, and among the 32 samples, 18 samples were found to be infected with Alternaria sesame. That is, 56% of samples were shown symptoms of Alternaria sesame. And among these 18 samples, the sample with highest percent seed infection was selected for conducting further studies on detection, location, and transmission. For conducting detection studies, standard procedures wise, standard blotter method, agar plate method, test tube water agar method were followed. For location studies, component plating method was followed. For conducting transmission studies, under seed to plant transmission, paper towel method, sterile soil method, that is nothing but pot culture method, and for proving plant to seed transmission, artificial inoculation and natural infection methods were followed. These are some photographs related to the study in standard blotter method. Among the 3 detection methods we have tried, highest percent recovery of the fungus was observed in case of agar plate method, followed by test tube water agar method, and then standard blotter method. This is pure culture of the fungus, Alternaria sesame. In case of paper towel method, we can see the infection here on seeds itself, and infected seedlings also we can see. And these are some photographs from pot culture studies. You can see the infection on cotyledons and defoliation of cotyledons due to infection. This is the symptoms, the characteristic symptoms of Alternaria sesame on cotyledons. And so these seedlings are at the age of 3 weeks. We have recorded the data, and these photographs indicates the infection on artificially inoculated capsules and also naturally infected capsules. Okay. And the data indicates that so for detecting seed-borne nature of Alternaria sesame and sesamum seeds, we have followed 3 methods, as I told. Among the 3 methods, agar plate method was found to be effective in recovering Alternaria sesame from the infected seed samples with 25.56% of seed infection. Okay, followed by test tube water agar method and then standard blotter method. While conducting an experiment on location of Alternaria sesame, the seeds were soaked for about 4 hours under room temperature conditions, and then they were dissected to different seed components by seed coat, pericarp, endosperm, and embryo. So when these were incubated under at 25 plus or minus 2 degrees centigrade in an incubator, we have noticed the highest percent recovery from the seed coat, 18.5%, followed by pericarp with 11.5% and endosperm with 16.5%. Whereas all the embryos were found to be free from Alternaria sesame infection. While conducting transmission studies, we have performed the experimentation with the seed to plant and plant to seed infections. And the seed to plant infections were carried out with the paper towel method and sterile soil method, that is pot culture method. So for all these methods, 400 seeds were evaluated, and among these 2 methods tried, paper towel method was found to be effective in giving more recovery of the pathogen, and also more number of seedlings have shown the symptoms of Alternaria sesame and cotyledons. With 23.38% of seedlings have shown infection in paper towel method when compared to sterile soil method, that is pot culture method, where 17.5% of infection was noticed. So in case of plant to seed transmission,
Under artificial inoculation conditions, the spore suspension with 5 into 10 to the power of 5 spore load was inoculated onto the healthy capsules, which were for which the inflorescence was covered and allowed for seed set. And after capsule formation, the healthy capsules were inoculated with the spore suspension. And later on, at the time of maturity, these capsules were collected and observed for the presence of symptoms. And this method resulted in 39.25% of infection, whereas under natural infections, where the field was left for natural infections, was recorded with 25.13% of infection. Okay? With all this data generated, we can conclude that from detection of pathogen on sesame seeds and its recovery from seed coat, it indicates that it's externally seed-borne nature, and recovery of pathogen from pericarp and endosperm indicated it's externally, it's internally seed-borne nature. Sorry, it's internally seed-borne nature. And symptoms on cotyledons while conducting the pot culture studies and also paper towel method indicated its seed to plant transmission and detection of pathogen from seeds collected from the infected capsules under both artificial inoculated conditions and natural infections indicated its seed transmission. So likewise, the seed-borne nature, that is externally seed-borne nature and internally seed-borne nature of Alternaria sesame in sesame crop was confirmed. That's all. This is the results of my small piece of study.
Thank you, madam. I think you should not publish this paper. It was 2-3 years over.
Yes, sir. This year we are publishing. Before that,
Okay, all the best. All
we
the
are
best.
preparing working sheet for
It's a very good work. It's a very good work. You must publish it. Thank you. Now we will. I think I request our co-chairman, Madam Chitrapu Ruth, to conduct this poster presentation session. Madam Chitrapu is there?
She is not there, sir.
Okay, then poster. Let us go. 5 poster presentations there. We will give only 5 minutes for each posters.
Yesterday, actually 1 minute, sir. Yesterday they submitted little bit late, sir. If they need other people, if they are in the meeting, if they want to present orally, you can give the opportunity, sir.
Okay, no. Okay, how many are there? May I know? There are 5 people. Kurla Sharad Babu, then Narsim Rao, then Kiran Kumar, Deepika, then Akshay Kumar. There are 5. How many
Sharad
of
Babu,
you are
can you
there?
speak? Sharad Babu is there.
Yes, sir.
Can you
Sharad
present?
Babu, you have a presentation.
Yes, sir.
Only
Yes,
3
sir.
to 5 minutes. 3 to 5 minutes. No, it's a poster.
Okay, sir.
Yeah, please, sir. Okay.
Kavya, yourself also, you also registered for no? Kavya BS.
Sharad Babu. Sharad Babu Kurla, yes.
You see, sir?
It's not visible. Yeah. Yes. Good.
Can you? It's audible, sir.
Yeah, audible. Very much audible. You can go ahead.
Okay, sir. Chairman of the session, and myself, K. Sharad Babu, Agricultural College, Bapatla. I'm here present my presentation on prevalence and molecular characterization of tobacco streak virus and virus. Majorly, groundnut is an annual oilseed legume crop grown in diverse environmental conditions. Tobacco streak virus causing peanut stem necrosis disease and groundnut bud necrosis virus causing peanut bud necrosis disease is a major constraint for nut production, especially the major concern for especially nowadays. So previously there is not survey conducted against these viruses. So attempts have been made for identification of prevalence of these 2 viruses in Andhra Pradesh. Of disease incidence, roving survey was conducted. based on percent disease incidence, number of infected plants to total number of plants. Based on typical symptoms, the suspected leaf samples collected from field and firstly, those samples for doctor, the positive samples confirmed again with RT-PCR by using gene primers. This is the map showing different locations, different GPS locations. Has been samples taken from different areas in 12 districts of Andhra Pradesh during Kharif and Rabi. This first one is the Kharif map showing GPS locations. This one is the Rabi map showing GPS location. The TSV is Kharif to Rabi, whereas in case of PB and D, bud necrosis disease, disease incidence more in Rabi compared to Kharif. During survey, I observed typical symptoms of TSV on leaf sample, on leaf like necrotic, small necrotic lesions on leaf, drooping of leaf, axillary shoot proliferation, peg necrosis, and pod necrosis. Majorly observing survey for TSV. In case of TB, symptom for the on weak part silvering of leaf as I earlier told for this year.
Sarath Babu, your voice is not clear.
Voice is breaking.
Your voice is breaking, Sarath Babu.
Come to the conclusion.
Okay.
Sarath Babu, go ahead.
No, sir?
Yeah, okay, go ahead.
Okay. The one of the isolate maintained copy and got the local lesions 3 to 5 days. Observed 3 to 5 days base period for TA and to CBI. Further, the presence of TSV and GBNV confirmed using it 7 for GB30.
Okay.
Further, late with different from India, other country isolates. TSV isolate clustered with our Indian isolates closely clustered with the ground. Event cleared other and the outgroup are CMB separate.
It's gone. Sarath Babu.
Sarath Babu, unnava? Sarath Babu.
No, it's cut, I think so. The next person, I think the next one, anyone?
Sir?
Okay,
yeah,
your
Sarath
screen
Babu,
is not there. What happened to you?
Your PPT.
Sir. It's okay, sir.
Oh, I think some internet problem with you. Sarath Babu, I think you can conclude because you already talked about everything. You just conclusion points you can just tell.
Okay.
You need not to share the screen. You can come to
my. Yes, sir.
Yeah. Something problem with your internet, I think so. It's not good. You don't have good bandwidth probably. Yeah, it's coming. You can conclude,
Okay.
Yes, sir, I'm there.
Yeah, I think Sharad Babu, I think you have concluded, no? Yeah, now Deepika, I think you can go ahead. Deepika?
Yes, sir.
Yeah, please present.
Yeah, thank you, sir. I think Sarad Babu has again appeared, sir. So
Yeah,
No,
you start.
your slide is visible. Deepika, you can go ahead.
thank you, sir. Just okay. So good evening to all of you, honorable dignitaries, participants. So in the morning, we had a wonderful lecture by Professor Manoharachari, sir, where he mentioned sex and might induce diseases among the emerging new disease. So here I am in relating to same. I'm here to present my work on effect of Seria lychee infestation on the volatile phytochemicals in lychee chinensis zone. So lychee is a subtropical plant belonging to the family Sapondaceae, and India is the second largest producer of lychee, China being the first. In India, it is mainly cultivated in Bihar, West Bengal, Uttar Pradesh, Uttarakhand, Jharkhand, Tripura, Assam, and Punjab. So lychee is an economically important crop for our nation, as India ranks among the top lychee exporting countries. But the production of lychee is by this mite, Asaria lychee, which infests the lychee leaves, and in serious conditions, the infection spreads to not only the flowers but the complete inflorescence and the fruits as well. So over here on this slide, you can see a lychee tree showing its GPS location of plant sample was collected. Then the second image shows a normal, and the third image shows an infected leaf where the has led to the formation of blister-like gall. Shows experiment. What I have done is I have compared the volatile phytochemicals, normal and
No, your voice is breaking, Deepika.
my voice is breaking, sir.
Yeah.
Should I be a little slow, sir?
Not slow. You can straight away tell what you want.
Okay. So I prepared the methanolic extract using dried powder of normal and gall leaves using Soxhlet apparatus, and then went ahead GCMS analysis. So here are the results of my GCMS analysis. Sir, is my voice still breaking?
No. Go ahead. It's clear.
Yeah, thank you, sir. So here are the results. I have mentioned only the significant compounds, though there were 43 phytochemicals detected in the normal leaves, and the gall leaves showed presence of 42 phytochemicals. So here are the important ones. Alpha methyl manofuranocide, vitamin E, and phytol. The amount of these three reduced from normal leaf to gall leaf. As you can see, that alpha methyl manofuranocide decreased from 72.49% to 60.82% in the gall leaf. Vitamin E2 reduced from 1.8 to 0.4%. These are the area percentages. Phytol reduced from 2.88 to 1.83%, though the caryophyllene increased from normal leaf to the gall leaves. Then betulinaldehyde and gamma cytosterol, two important chemical compounds, were found absent in the gall leaf. And betulinaldehyde has an anti-tumor property. So you can see it was absent in the gall leaf. Beta longipenine and lupiol, though, were found to be present only in the gall leaf and they were absent in the normal leaf. So here you can see the graph which has been shown to again to show the area percentages of some of the significant compounds. And these two chromatograms are basically of the normal and the gall leaf. So from the above results, we can conclude that some of the important compounds were affected by the mite infection. As I previously mentioned, that some of the compounds decreased in the gall leaf, some were found to be increased. Then there were compounds like betulinaldehyde and gamma cytosterol, which were completely absent in gall leaf, and few others were present only in the normal leaf or only in the gall leaf. So in a study like this can basically give an insight the biochemicals being affected due to the host plant and the mite interaction, and they can help us to basically prepare strategies for the management of the mite so that this economically important plant is not affected because of this mite. So thank you so much, sir, for giving me
Thank
this
you.
opportunity.
Okay. Thank you, Deepika. Okay, it's a good work on a lychee crop on mite infection, and this it's very good. So let us go to the next one. You can close this.
I'm closing, sir. I'm closing.
Yeah. Next one, Kiran Kumar. If Kiran Kumar is not there, Akshay Kumar.
Yes, sir. Akshay, I'm here, sir.
Akshay Kumar, you're since beginning you're there. Please go ahead your presentation.
Yes, sir.
Oh.
Is my screen visible, sir?
Yeah, visible.
Good evening, one and all present over here. Myself, Akshay. I'm an PhD student at the Indian Agriculture Research Institute. Today, I'm going to present a poster on my research topic, identification and utilization of simple sequence repeat markers for identifying and studying the genetic diversity among the Fusarium graminearum isolates, which causes head scab disease of wheat in India. We all know that wheat is a major staple food crop on which the more than 2 billion people worldwide depend on. And this Fusarium head scab, it is caused by Fusarium species complex, which worldwide causes around more than $10 billion loss. It also produces some of the mycotoxins such as DON, ADON, and zearalenone, which will be leading to the feed refusal and many illnesses in animals, poultry, piggery, etc. And coming to Indian scenario, right now this is not in such a major disease, but this disease already started appearing in a severe form in many areas of cooler and hilly regions. And due to climate change, this disease is going to be a future threat to the Indian wheat production. Despite of the sufficient knowledge available on the genetic variation of Fusarium graminearum in the world scenario, but in Indian scenario, a very limited literatures are available on this pathogen. So with this lacuna, we formulated our objective as to study the genetic variation among the Fusarium graminearum isolates causing Fusarium head scab or head blight of wheat in India. We collected isolates, we collected around 110 naturally infected wheat samples from Nilgiris of Tamil Nadu and Laul Spiti Valley of Himachal Pradesh, and also 2 isolates which were already deposited in Indian type culture collection in New Delhi. And out of this, we could find only 29 isolates as Fusarium graminearum. These were identified by Indian type culture collection, and later on, these all 29 isolates were tested for pathogenicity on the susceptible variety HT. On the susceptible variety. Later, these fungus was cultured in PDB broth, and whole genome DNA was isolated, and that DNA was tested for ITS regions, and we amplified that ITS region, and we sequence that, and we analyze for similarity analysis. Later, using the already available PH1 whole genome sequence in the database, using a MISA tool, we identified the SSR markers in the Fusarium graminearum genome, and later on we selected 23 markers such that it represents all the 4 chromosomes of the Fusarium graminearum. And we designed primers and validated them for all the 29 isolates. Coming to the results, in morphological characterization, we studied different characters such as color of mycelia, which varied from light pink to the brownish pink, and the spore size, that is for macroconidial size. Also, we studied it. The length varied from 43.6 micrometer to 67.1 micrometer, with the width varied from 5.2 micrometer to 10.08 micrometer. And coming to the growth, Wellington isolates were found to be growing at much faster rate compared to the Laul Valley isolates, and these isolates produce the pink pigmentation on the PDA media, and also in carnation leaf agar and potato carrot leaf agar. All medias it produced pink pigmentation. And coming to results of internal transcript space region amplification, we amplified all the genomes of for all the isolates for ITS, and we sequenced them. We could find percent similarity with Fusarium graminearum with 90, starting from 98 to 99.45%. And using MISA tool, we identified the number of SSRs present in the Fusarium graminearum genome. We could find that around 1,577 markers were identified, out of which majority were XR repeats, that is of around 57%. It comprises errors of around 893 markers, followed by try, that is 17%, and the least were found to be tetra repeats. And among them, we selected 23 regions, and we designed a primers for them, and validated those markers in all 29 isolates. Then we scored those markers, and we scored for number of alleles, but we could two SSR markers didn't amplify anything in all the isolates, but these remaining 21 markers could amplify around 34 alleles, and the SSR 21 which amplified around 4 alleles, which 4 different base pairs, and the PIC values of the gene diversity, what we calculated, it ranged from 0 to 0.97, indicating that there is much scope for these primers to study the genetic variation among these isolates. And later on, using Jacquard coefficient, we construct an UPGMA dendrogram. All the 29 isolates clustered into 2 different clades, and FGW27, FG and FGL2, and 2 different isolates from 2 different regions were grouped together in clade A, and all other isolates were grouped together in the clade B. We can conclude from this that there is an existence of within region, and there is an existence of within region variability also. And coming to the conclusion of the study, we collected around 110 samples, out of which we could find only 29 Fusarium isolates. Others were different Fusarium species, and these isolates were characterized for cultural characters, and also tested for pathogenicity, and all isolates were found pathogenic. And in Fusarium graminearum whole genome, we could find around 1,577 SSR markers, out of which XF were more, followed by try repeats, and 21 markers in this study amplified 34 alleles in Fusarium graminearum, all isolates, and PIC values range from 0.0 to 0.97, and dendrogram analysis yielded 2 major clades for us. And this data, what we obtained from study, enrich the current Fusarium graminearum genetic variability information and provide good foundation for better understanding of within the region and among the different regions of India, what the variability exists. And at last, I would like to acknowledge IRI and ICR and Indian Institute of Wheat and Barley Research and IPS for granting me this opportunity to present my piece of work here. Thank you, one and all.
Okay, thank you, Akshay Kumar, for a nice presentation on this particular pathogen, Fusarium graminearum, and it's very good work what you have done, and it's very nice. The next one, anyone is there? I think with this it's over, Dr. Sesagran. And if our coach chairperson is there, she can give.
Tulasi is available, sir.
Huh?
Tulasi is available, sir.
Tulasi, your list is not there. Okay, you can go ahead. This is the last presentation. Okay. Sesa is the last.
Yafat Shahnaz, you also registered, no, yesterday. Are you there, Shahnaz?
Shahnaz is
yes,
there, but
yes, sir, I'm here.
Okay, can you present? Afterwards, you speak in.
No, sir, I'm not ready for today.
Okay,
leave Actually, it. I did not have the exact time.
Okay, okay. Who is that person?
Can I present tomorrow, sir?
Okay.
Okay, thank you, sir.
Tulsi Bhai.
Yes, sir. I am here, sir.
Tulsi, okay. Present.
Okay.
Only 3 to 5 minutes, not more than that.
Okay, sir.
It's only poster, you know.
Good evening to everyone. Impact on endophytic bacterial metabolites on post-harvested management of tomato. My name is Tulasi. I'm pursuing PhD program under the guidance of Dr. A. Amrutavalli Madam, Department of Microbiology, Acharya Nagarjuna University, Guntur.
Go for slideshow, please.
Okay, sir. Abstract. Nowadays, fruits and vegetables highly demand in market in nutrition and value. Fruits, vegetable has short shelf life due to its nature, and it's under effect and damaged by insect microorganisms. PR post-harvest condition play major role in the quality of product and post-harvested preservation of fruits and vegetables in big challenge for the world. present in the aim to evaluate the pre-harvested treatment of tomato with plant growth promotion endophytic FR3 and FR19 on quality of post-harvested tomato in terms of nutrition and value apart from the growth promotion. UV and IR spectral ethyl acetate of FR3 and FR19 clearly suggested the presence of fungal cell walls enzymes and coated with ethyl acetate extract of FR3 and FR19 showed the shelf life to an extended for 24 days with significant interaction of carbohydrates and proteins. Introduction. Tomato is one of the most popular
So come to objectives. Come to objectives. Yeah, present the objectives. Yeah.
To study the efficacy of endophytic PGPB on plant growth promoting harvested tomato health by pre-harvest inoculation and evaluated to the ethyl acetate coating on extending the post-harvested quality contributed to tomato Lycoperdicum esculentum mille by post-harvested coating of extraction ethyl acetate extract by endophytic bacteria. Materials and methods. Antagonism of endophytic bacteria against the Fusarium oxysporum. Effect of endophytic bacteria strains on plant growth promotion. Preparation of bacterial cell suspension, seed treatment and nursery experiments. Extraction of crudes, UV visible and FTIR spectral lines. Quantitative estimation of metabolites. Design experiment and statistics analysis. This is antagonistic activity of Fusarium oxysporum and my endophytic bacterial Bacillus FR3 and FR9 show the antagonistic effect of Fusarium oxysporum. Zone of inhibition control 0% FR3 is 54.44%, FR9 is 65.97%. This is UV spectroscopy of ethyl acetate. FR19 is 23 peaks in a UV visible spectra. A major peak is observed in 244 in observance. FR19 is a spectroscopy of UV visible in peak observed in 250 nanometers. This is the FTIR results. My peaks is maximum observance in 655. This is mainly amino acid peptides are present. This main group of ethyl acetate extracts of crude extract. IR spectroscopy of results. Spectroscopy analysis showed that the purified substance structure lipid observance value 3350.34 cms indicated the presence of NH groups in the molecules. Observance value in 2009 1415 demonstrated typically CH3 stitches vibration in the peptide chains. Observance value appear to the 655.415 and 1535.437 same where stitches vibrate to C double bond and CN groups in peptide bonds maximum. Tomato fruits coated with ethyl acetate extraction of FR3 and FR19. Ethyl acetate coating results. Those tomatoes that are coated by endophytic bacterial isolers have shown resistant to pathogenic fungi, Fusarium oxysporum, and that are not coated so diseases. Ethyl acetate crude extract proteins, carbohydrate drops.
Okay.
Conclusion. The edible coating is the most efficient method for enhancing the post-harvested quality fruits and vegetables. The edible coating is effective technique to preserve the fruit and vegetables. It does not create any harm to the environment and can act the oxygen moisture barrier, thus delaying the fruits from ripening. The herbal coating is the developing technology that has antioxidant, antimicrobial and antifungal properties. The herbal extract consists of vitamins and minerals. It can also impact towards the nutrition value of edible coating. Properly, the herbal edible coating will be to the alternative methods for the wax and chemical coating in the fruits and vegetable. Post-harvest management of fruits and vegetable is major for the all nations whose backward to agriculture. In this view, we have discussed general edible coating along with the edible coatings which would have a wide prospect in the coming future for maintenance of post-harvested quality.
Okay. Thank you, Tulasi. Thank you, Tulasi, for presenting very elaborate work for this poster. And I think with this we conclude.
Thank you, sir.
Yeah, with this we conclude. So in this session, there were 3 lead papers and 10 oral presentations were there, and 4 poster presentations were there. So it's all very good presentations were there, especially in the lead talk. There were 3 by Dr. Hema and Dr. Amit, then Dr. Amruta. They gave very wonderful presentation on 3 different topics. One is on phytoplasma, and another was on virus disease diagnosis, and third one was endophyte, and all from 3 different area. It was very nicely they covered this particular talk on diagnostics and detection as well as characterization of plant pathogens. And it's very wonderful because they covered all 3 different pathogens, and it would be very useful to all the students, plant pathology students, especially the masters who are interested in doing research. They get good exposure with the 3 good lead speakers picked up by this IPS zone. And coming to the oral presentations, they were very good presentations on different topic, again virus as well as fungal and bacterial. Of course, the number of papers on bacterial diseases were very less. Maybe 1 paper was there, I think so, but otherwise so on seed and other papers on soybean and all was there. And again the paper on sunflower necrosis disease was very good, and again sugarcane yellow leaf virus was very nice by Parameswari. Then the young students, like master students like Chandana, presented very nicely about the this gummy ooze secretion and bottle gut disease, a new disease. And of course we got able to listen, though it is a central zone, we could be able to listen from this Kashmir, that also from Assam, and also I think from Chhattisgarh comes under the same group, no? Yeah, it was very good. The different people could able to give and share their presentation, including the last one by Fusarium graminarium, the poster that also was very good by Akshay Kumar. So overall, this session, the presentations were very nice, and I hope our rapporteur has taken note of all the question and answer session which was there.
నేను డిపార్ట్మెంట్ లో ఉన్నాను, నన్ను హాస్టల్ కి తీసుకెళ్లరా?
Hello. Okay. So that's what. It's very, very wonderful session on this. In fact, around 45 to 50 people participated initially, and now around 25 people are here listening to our the talk. Overall, this session was very good. Dr. Robin Gogai for joining now. Dr. Robin, oh, Robin could not hear. Dr. Robin Gogai, our secretary has joined. Okay, so it was very nicely arranged by Dr. Srinivasulu Garu and as well as Sesakaran. This particular session was really fruitful and carried a wide range of topic, both lead as well as oral as well as poster presentation. And I hope that we will be giving the best oral presentation as well as best poster presentation probably at the end of the tomorrow's validity session. So I thank I think I missed our co-chairperson Dr. Chitra Purut, and as well as our Vijay Lakshmi Garu also there or not, I don't know. But anyhow, I think she has taken note of all those the proceedings taken place. So I would be giving tomorrow in the validity section all the recommendations from arose from this discussion of this session. So I once again thank the president of the central zone, and as well as the councillor of central zone for this IPS zone. And I also thank Dr. Robin Gogai, the secretary of IPS, and Celia Chalam. Celia Chalam also, since beginning, she is there listening to all the talks, and it's very good. And I thank all the organizers for giving me this opportunity to share this important session. Thank you very much, Dr. Srinivasulu and Sesakaran. Once again, anything
Welcome,
to
sir.
Yeah, I
We are very
conclude
happy
this.
to see you, have
We
you
will
here.
announce the award. I will send the award list and the proceedings tomorrow to you for the validity session.
Okay, okay, sir.
Thank you, Dr. Salvarajan, for your excellent conducting of these proceedings.
Thank you.
And for your cool and calm.
Yeah.
Especially.
We want to listen more.
And also,
I didn't get upset when somebody asked that I want to present. I really appreciate that.
Yeah, that's why, madam. That's why he is a very good person, very calm and very cool. He's a suitable person for the chairman, and he has done excellently his job. Really, thank you, Salvarajan, for accepting and for conducting excellently.
Thank you.
Thank you.
Thank you. So tomorrow validity session we have to give your.
Tomorrow valedictory, all the chairmen has to give their recommendations and a brief report.
Kindly
Now, I
ask
request Sheshakaran also please inform to all the chairmen of the committee during the validity session. They have to prepare a brief note about the recommendations and number of participants, whatever they may be, and they can give a presentation also.
Yeah.
Okay, okay, sir.
Ravin Gogai, sir.
Selvarajan, sir, tomorrow you attend other sessions also, sir.
Any
Krishna
remarks
Reddy, sir,
of
is
our,
the chairman is there.
No, I will come for that validity.
No, sir, other sessions also you can join, sir. No problem, sir.
Sheshakaran, our secretary Ravin Gogai joined. So
Yes,
you can take remarks from him also.
Yeah.
So this is definitely end of this first day of your 2 days program. Is you have really very nicely conducted. So what actually I want to mention here is right from the southern zone. Actually, southern zone started with a very good pickup.
Okay.
And after seeing their performance and organizational setup, all other zones also get received encouragement. What I feel. So that's why each and every zone really very nicely organizing, and this is remaining no more as a zonal. It has become international.
Yeah.
Every zone is inviting many people from outside our country also.
Yeah.
And the system, particularly this virtual system, made really the entire world in a very closer distance, hardly half a meter distance, but we can talk each other, and there is no any time difference. You see, in the morning time, Dr. M.S. Reddy was talking in the night time from his country, and we are able to listen within a fraction of a second without any deviation of timings. So really, this system has benefited us. So of course, both of you, Dr. Srinivasulu and Sesakiran, they have sincerely worked and set up the whole program. And one more thing I have been observing so far today, this is the 5th zone for this doing this journal chapter. One person, Dr. R. Sridhar from Kotak, but today he is missing, and Dr. C. Manoharasari, and another one, R. Silvarajan. These 3 persons are really all along they are attending all the journal chapters. And this has become very possible only because of virtual system. It's very nice.
Yeah.
And all the participants nicely showing their interest to join, present their work. So this is only the platform to express their views, experience, and also what they are doing, they are able to see. Otherwise, it is really very difficult because silently everybody is working and they are sending their research finding to some journals and publishing very silently. There are only few people are accessing, but here, lively, we can have their experience and their suggestions, even our suggestions also they are receiving. This has become really a very fruitful function. So tomorrow also definitely I think I have to keep my this some concluding remark. But first the concluding remark is that okay, it is very nicely done, and we hope tomorrow will be very, very actually vibrant because there are so many competitions are there. So we are waiting for the tomorrow's programs. Okay, thank you once again.
Thank you, all and all.
Thank you. Bye.
We can end the meeting, sir.
Celia.
Yes.
Thank you, ma'am, for attending this one, all the sessions, Celia madam, Gogoi sir. I should thank on behalf of my Central Zone, Selvarajan sir, for acting as the chairman and doing all this great work, sir. You please attend the tomorrow's session also, and also valedictory function.
Yes.
Sure.
It's our invitation from our organizing committee.
Okay. Thank you. Bye.
Okay, thank you.
Okay, I'm leaving. Bye.
Thank you.
Okay, sir.